So good afternoon, everybody, and welcome here in Geneva. My name is Marlène Bras, and I'm the director of the HIV programs and advocacy department at IS, where the Global HIV Vaccine Enterprise sits. It's a great pleasure to open this year's annual stakeholder meeting and to welcome you all in Geneva. For those of you who are in the room, meaning that we are around 20 people in the room and we expect 60 people online joining us from various places in the world. So this year is a very special year for the enterprise as we celebrate the 20th anniversary of the enterprise. It was in 2003 that a broad group of experts and thought leaders in the HIV vaccine fields met and proposed the creation of a policy forum that later became the Global HIV Vaccine Enterprise. Although an HIV vaccine remains elusive, with still many challenges and hurdles, much has been accomplished. And there are reasons for celebration and optimism about the future for HIV vaccine research. So today's meeting is an opportunity for all of us involved in the HIV vaccine R&D to come together and reflect on today's challenges, identify way forwards, and determine how the vaccine enterprise can support a global effort towards an HIV vaccine. As tremendous progresses have been made in the past decade with ARV-based HIV prevention, this meeting is also an opportunity to reimagine a much-needed advocacy strategy for HIV vaccine R&D. So the first part of this meeting will consist of 2 presentations, followed by a roundtable discussion. We will then have a short break and move to the second part of the meeting with a presentation, followed by a second roundtable discussion. And as a reminder, as we always do, there's few housekeeping rules that I would like you to keep in mind. The first one is for you who are in the room, whenever you want to talk, please press the little button that you can see in front of you so our online participants can hear what you are saying. The meeting will be recorded, so the recording will be made available to the participants and on the IS Plus platform as part of the educational materials after all the necessary consents have been acquired. By staying in the meeting, that means that you are consenting to being recorded for this portion of the roundtable. For those of us who are joining us online, welcome. It must be maybe early morning or early evening for you, depending on where you are when you are basically. But we encourage you to send questions to our presenters at any time through the Q&A function at the bottom of your screen. They will be addressed during the roundtable discussion and through this meeting. We kindly ask all virtual participants to stay muted and with their camera off through the duration of the meeting. And if you have any question that you would like to pose during the panel discussion, please raise your hand and unmute yourself. For those of you joining in person, we encourage you to be very active. We want that to be very lively and very active. So please don't be shy and don't hesitate to share your ideas and thoughts. So we're going to start with the first part of this stakeholder meeting. And the first part is dedicated to identifying and addressing the scientific gaps of HIV R&D. And our first speaker is Vincent Muturi-Kioi, who will present on the state of the art of current approaches. Vincent is a senior medical director with the clinical development team at the International AIDS Vaccine Initiative Kenya. He focuses on discovery medicine trials for HIV vaccines, and his presentation aims to provide a bird's eye view on the current immunogens and vaccine strategies, their status, and expected next steps. The presentation will include less discussed and less publicized approaches. So, Vincent, the floor is yours.
Thank you, Marlene. Can you hear me?
Yes, thank you.
I believe Julia will be sharing my slides. I can't see them at this time. All right, thank you. So thank you, everyone. Thank you to the team at the HIV Vaccine Enterprise for having me here. Next slide. I'd also like to thank all those who make our work possible and all our donors. Next slide. So this is the outline of my talk. I'll be speaking for the next 15 minutes and highlighting the persistent burden of disease caused by HIV, challenges associated with the development of a vaccine against HIV, and as Marlene said, some of the strategies for vaccine development that are currently being undertaken. Next slide. So with regard to the burden of disease, I think it's important that we all recognize the fact that a lot of progress has been made in the combat against HIV, but we still see a very unacceptably high burden of disease around the world. According to the UNAIDS estimates, since the beginning of the pandemic, more than 39 million people have been infected. And as of 2022, we were still seeing more than 1.3 million new infections being reported annually. The number of deaths is still unacceptably high, even with the access to effective treatment, more than 680,000 deaths estimated in 2022. And from the perspective of the situation in Africa, we still see 60% of new infections from the continent, with a disproportionately high number of infections suffered by adolescent girls and young women. Next slide. The graph on the left of the screen does highlight the fact that we missed our goal to reduce new infections to below 500,000 cases by 2020. And the current trajectory of infections still highlights the fact that we are likely to continue to miss the set goals with regard to elimination of HIV as a public health threat. There are multiple risks that we are still keen to consider as we move forward with trying to end the pandemic. The demographic changes, especially in sub-Saharan Africa, with the increasing population of young people who would be at risk, and with this growth in population, without a change of incidence, we risk losing the gains that have been made in combating the disease. In addition to that, the risks to the programs that have made treatment accessible, and the fact that with reduction in funding to these programs as a tangible risk, there is a risk that we might see reversals in the future if the efforts to maintain care and treatment programs at the level that they are and increase access are not maintained. The risks of drug resistance persist, and the question of whether we can develop new products to combat this remain. Next slide. So why a vaccine? Vaccines would give us the opportunity to have an intervention that provides long-term protection beyond the time when it's administered, possibly for years and preferably for the rest of an individual's life. The opportunity to have a product that requires fewer interactions with the health system would make it an accessible intervention for those who need it most and who might have limited opportunities to access health programs. The opportunity also to access to populations in the most remote parts of the world where the interventions against HIV are needed most would also ensure that a vaccine would drive us towards our elimination targets. Next slide. So for the next few minutes, I'll highlight where the challenges have been and what we think will be needed to develop an effective vaccine. Next slide. Many of you are aware of the fact that traditional vaccine approaches have failed, and this is mainly due to the characteristics of the virus we are fighting. HIV is hypervariable, results in a persistent infection that lasts for life in all those who are infected, and it continually evades the natural immune responses that are directed towards it. In addition to that, HIV attacks the immune cells that are important for generation of an adequate immune response and makes it difficult to ensure elimination of the virus. And lastly, when it comes to the development process, the commonly available animal models have proved insufficient for the development of a HIV vaccine, as they rarely predict what we see when it comes to how the vaccines work in humans. Next slide. For the strategies that we think would be successful, it is likely that we will need to stimulate both the humoral and cell-mediated immune responses to effectively prevent or effectively prevent HIV infection or drive viral suppression in those who are infected. Next slide. When thinking about the HIV vaccine efficacy trials that have taken place so far, what we've seen is that an empirical approach has been taken to vaccine development that has resulted in the majority of these trials not demonstrating an efficacy endpoint. The RV144 did find a signal of efficacy, but there was failure to replicate this in subsequent trials using the same concept. Next slide. I think I've already highlighted the point that we would need to have generation of appropriate or effective immune responses targeting the humoral and cell-mediated arms of the immune system. And for the humoral responses, it is likely we will need neutralizing antibody responses that have not been generated by any of the vaccines tested in efficacy trials so far. Next slide. In addition to that, we have seen a move from empirical approaches to vaccine development to rational approaches. And what does this mean? In previous attempts, we have tested vaccine candidates with the hope that we will see an efficacy signal that allows us to determine correlates of protection thereafter. And this has meant that we would have to carry out large trials in order to determine which vaccines work and therefore move back to perhaps either improve those responses or modify the concepts for better responses. Using rational approaches, we start from the end goal with the aim of ensuring we have a correlate of protection identified, and then creating a strategy to elicit that what we believe will be a protective response before moving into later stages of development. And where we are now is conducting multiple experimental medicine or discovery medicine trials that will allow us to determine which vaccine candidates are able to elicit those immune responses that we believe will be protective and advance those most promising candidates towards efficacy trials once we have a regimen we believe would be effective. Next slide. So I'll start off with vaccines that aim to induce broadly neutralizing antibodies targeting the humoral immune response. Next slide. There are several strategies to try and generate broadly neutralizing antibodies. There's lineage-based vaccine designs, which are based on immunogens that mimic sequences that have been isolated from individuals followed longitudinally in observational studies that have gone on to develop neutralizing antibodies themselves from natural infection. There's the germline targeting vaccine design that aims to target precursor cells that would be with engineered immunogens that would activate these precursor cells and start individuals down the line towards developing broadly neutralizing antibodies. And there are immune-focusing vaccine designs that use immunogen sequences designed to focus the response to one epitope, but which are germline agnostic. Next slide. The work to develop broadly neutralizing antibody responses using vaccines has taken a lot of learnings from what we've seen in people following natural infection. And so if you go to the next slide, what we are hoping to do is target, and this highlights the germline targeting approach, to target naive B cells that have the characteristics to then progress and produce broadly neutralizing antibodies, we will likely need a sequence of immunogens to, number one, carry out the priming using the germline targeting, and then shepherd the immune response from those naive B cells that have been activated towards production of broadly neutralizing antibodies. This concept has already started being tested in the clinic. We've seen germline targeting immunogens work, activating those naive B cells, and work is ongoing to try and see if these immune responses can be boosted towards generation of broadly neutralizing antibodies. Next slide. Aside from those vaccines targeting broadly neutralizing antibodies, I must mention there are other concepts being tested for humoral immune responses to try and produce polyfunctional antibodies that would increase the opportunity for protection by generation of immune responses that are polyfunctional and use FC-mediated responses to help with prevention. For cell-mediated immune responses, we are aware of the fact that during natural infection, at the peak of viremia in acute infection, that the viral load is reduced to the set point thanks to the work of CD8 positive T cells. The goal in this case would be to elicit a response that allows suppression of that viremia, possibly below a transmission threshold, and essentially maintaining long-term suppression of viremia or prevention of infection. Next slide. There are multiple cell-mediated approaches that are either in the clinic or going into the clinic soon. We've seen mosaic vaccines that use computational designs of small sets of viral proteins with the aim of being able to elicit responses to the breadth of viruses that exist or that are circulating in the community. And in addition to that, we've seen designs of vaccines with focus on conserved regions of the virus that have minimal diversity across all forms of HIV. Similar to that, there are networked epitope vaccines which focus on mutationally constrained regions of the HIV proteome and aim to elicit responses that would be applicable to all circulating strains of the virus. Next slide. This slide highlights one of these concepts, conserved mosaic vaccine developed by Tom Hankey and the team at the University of Oxford. This has successfully been tested in multiple phase 1 trials, eliciting broad T-cell immune responses and demonstrating that there were high magnitude of responses both in individuals enrolled in European countries as well as in sub-Saharan African countries. Next slide. I'd like to summarize my talk by highlighting the fact that HIV continues to cause a high burden of disease around the world. The demographic changes that are occurring, especially in sub-Saharan Africa, coupled with ongoing high incidence in specific groups, including adolescent girls and young women, could reverse the gains that have been made in epidemic control and cause us to miss the mark as far as elimination of HIV as a public health threat. A vaccine used together with other available interventions could help end the pandemic. And as we are aware, traditional approaches have failed to develop an effective vaccine. A successful strategy will likely elicit broadly neutralizing antibody responses and cell-mediated responses, and there are several promising strategies that are currently in development. Thank you very much for your attention.
Thank you very much, Vincent. So our next speaker is Sheila Balinda, and Sheila will present on mRNA platform for the development of an HIV vaccine. Sheila is an assistant professor and senior scientist at the Medical Research Council, Uganda, Virus Research Institute, and London School of Hygiene and Tropical Medicine, Uganda Research Unit. The presentation of Sheila will provide an update on current and planned studies, using the mRNA platform. Sheila will discuss its value and limitations, with special attention given to the imperative on ensuring broad accessibility for research endeavors. Sheila, over to you. Thank you.
Thank you very much for your kind introduction. And I would like to thank the organizers for the opportunity to present here today. Yes, as highlighted, the title of my presentation is Messenger RNA Platform for the Development of an HIV Vaccine. I'm almost afraid to say that other part, but however, should we be curbing our enthusiasm on this front? Next slide, please. A brief background, as you're all aware, the history of HIV vaccine efficacy trials has spanned over 20 years, approximately 21, since they started in 2003 to date. And so far, as highlighted by our previous speaker, we have not yet succeeded. Most of the candidate HIV vaccines have been designed using a combination of traditional approaches, such as the protein subunit, viral vectors, and DNA delivery platform strategies. Next slide, please. During the COVID-19 pandemic, we did have a messenger RNA platform that rose to prominence. Work had been ongoing, but rather below the radar, and this was able to contribute significantly to public health intervention. In the US alone, you found that approximately 1 million deaths were presented by November 2021, and 10 million hospitalizations were also prevented at that time, with about 36 million people prevented from actually falling sick. And the key platforms that did rise to prominence were the one from Moderna and Pfizer technology. However, this work had begun previously about 1990, and this laid the foundation for a messenger RNA platform. It has been applied to several other infections, including influenza and all the listed viruses on the slide there. Next slide, please. Currently, because of the immense success that is attributed to COVID-19 messenger RNA platform, the HIV research world has also slowly evolved towards adopting this platform. And as Vincent has highlighted, we have the THIV ConserveX, which is led by Professor Tom Hankey and his research colleagues. It's currently at phase 1. It focuses on the bivalent mosaic, which is a third iteration of vaccine development with regard to that design. The goal here is to ensure that we get T cells that are produced broad and specific to specific conserved epitopes across different HIV subtypes. Then we have the messenger RNA 1644. This is by William Schaeffer and his team. IAVI is quite critical in this work, and it's a germline targeting approach. And then we have the 3 BG505 HIV trimers led by NIH team, Scripps, and all the other colleagues. And these are also in phase 1 clinical trials using messenger RNA platform. However, it is important to highlight at this point that unlike SARS-CoV-2, with regard to HIV, we see a lot of diversity. For example, by I think December 2022, for SARS-CoV-2, we only had about 20 variants, while with HIV, we are in millions of variants to date. And then secondly, again, with SARS-CoV-2, already the structure protein that will elicit a potent virus has already been identified. With HIV, we are still struggling to identify the most appropriate sort of immunogen. So these are some of the challenges that will also affect the current messenger RNA platform vaccines. Next slide, please. A quick recap with regard to messenger RNA vaccine structures. We have basically 2 platforms: the conventional non-amplifying messenger RNA. And here we just have the antigen inserted into the carrier, and this results in normal production of the antigen. On the other hand, we have the self-amplifying messenger RNA. And the unique thing about this platform is we have a replicase gene that is introduced ahead of the antigen. This results in much more antigen produced if this platform is used. Currently, most of the successful messenger RNA vaccines have been with a conventional platform, although most of the work that is ongoing with the self-amplifying messenger RNA is at preclinical level with significant data and results, particularly for the Zika virus. This was done by a Belgian group. And then, of course, we are using nanoparticles as a form of delivery. They are stable, ensure targeted delivery, and also stimulate TFH cells. Next slide, please. And then again, with formulation, there are several ways these viruses could be formulated. And key, or the standard gold currently, is the lipid nanoparticles. The reason I'm highlighting this is because this work is ongoing at the MRC in Uganda. And because we don't have this equipment yet, we are using the liposome approach as opposed to the standard gold. Next slide, please. The advantages of this platform, as we all know, is it's quick and therefore will allow us to test several vaccine candidates at almost the same time. So you have your plan A, plan B, and plan C. And then the ease and speed of production, immunogenicity makes it also very appealing. It enables the rapid synthesis of safe vaccines using cell and virus culture-free processing, making it much easier to work with. And then they can induce both B and T cell. And then with the LNP messenger RNA, we can then activate the T follicular helper cells as highlighted previously. Some of the advantages, of course, could be, next slide, please. Yes, could be allergies and of course, renal or organ failures but of course this is quite rare. And then in sub-Saharan Africa, we have of course cold chain requirements for messenger RNA vaccine delivery and accessibility, which may provide a challenge for some of the communities in that area. And then the third and most important is HIV diversity. That is just some work showing preliminary sensitivities of contemporary viruses from Uganda, specifically from the Good Health for Women cohort. We can see that at IC80, and then 0.0 gram per mil, most of the TF sequences were resistant. So we needed a significant increase in concentration for sensitivity. And then summarized at the bottom, those are the broadly neutralizing antibodies. They are sensitive too. Additionally, we've been involved in phenotypic characterization of the viruses in Uganda. And of course, they did follow the central dogma, transmitted viruses prefer the CCR5. However, what was interesting about these viruses is that they were all resistant to the co-receptor antagonists that we worked with at that time, as summarized by that point. Next slide, please. Our immunology team, led by Dr. Jennifer Seranga, has also been involved in collaborative broadly neutralizing studies with UKZN. And some of the data there shows that overall inhibition is higher in historical samples, about 7 of the 10 monoclonal antibodies, than in the contemporary viruses. So overall, PGT15 and VRC01.23 are the most potent inhibitors if you cut across historical and contemporary. And the clear differences are quite evident across subtype A and C. And this is the meaningful interpretation data with statistical significance that could be computed at that level, with key critical relevance gaps observed for subtype D, AD, and A, which are quite prevalent in the East African region. That is in Uganda, prevalent D, as well as Kenya and Rwanda around, and we see an increase in the contemporary viruses, we see an increase of the recombinants, which all introduces new dynamics for vaccine development. Next slide, please. And then briefly, I'll just give an overview of the messenger RNA platform that is being set up at the MRC in Entebbe, Uganda. This is funded by Ayavi. It's led by Professor Pontiano Kalebu and a PhD student, Paul Kato. Next slide, please. And here, as you can see, it's a collaboration between majorly 3 institutions, the UKZN, that is screening and selection of HIV/AIDS sequences that we are looking at from sub-Saharan Africa, and then the ATRP in India that is involved with HIV envelope trimer production and characterization. Again, MUL is involved in the messenger RNA synthesis and characterization, and we are using both available platforms, the conventional as well as the self-amplifying. And then immunogenicity assessment, this capacity has been built at the unit in collaboration with other groups, majorly in the UK. Next slide, please. The lab has been set up. The PhD student traveled through a collaborative arrangement to Imperial College and worked with Professor Robin Shattock. Upon his research, we did have infrastructure set up. Next slide, please. And then this is the summary pathway. I just wanted to highlight that currently at MUL, under HIV envelope trimer characterization, we've built capacity for ELISA and Western blot, although we look forward to also adopting SPR and NSN, as that would improve the quality. And we also hope to gain the formulation equipment that will help us generate vaccine candidates that are critical using the lipid nanopore particles. And then, of course, this is a summary of some of the preliminary data that is ongoing using the BG505DS SOCIP. Here we've generated several vaccine constructs using both the messenger RNA conventional as well as the self-amplifying RNA. And this has been tested using the native page for expression and then binding ability summarized in the left-hand panel of that slide. And we see this with PGT15. The summary progress is that training capacity has been completed. We've cloned this PG505 into the platforms, messenger RNA platforms, and then we have also synthesized this, and then assessing in vitro expression of self-replicating RNA and messenger RNA using that particular immunogen is actually completed. I was updated yesterday, apologies for that. And then we have currently started working in collaboration with India, encoding HIV trimers selected from the trimers within the sub-Saharan African cohorts. Next slide, please. The challenges and mitigation steps. Basically, we restricted ourselves here to technical challenges where we did have limited capacity for characterization of the HIV/AIDS trimers. However, through collaborative efforts with India, we've had the PhD student travel, and we are currently adopting this. And then the formulation equipment would also be quite key to ensure we use the gold nanoparticle for carrying the immunogen candidates for preclinical studies. And then, of course, as highlighted, animal models are a problem. So far, we've been working with the BALB-C for Rift Valley using this platform, and we've seen a lot of background. So we hope we will be able to move into probably the humanized mice to be able to evaluate preclinically the vaccine candidates on this front. I would like to thank IAVI, of course, for funding most of these studies, and I'm also grateful to the team at MRC who work day and night to ensure we make progress on this front. Thank you all for listening.
Thank you so much, Sheila, and thank you, Vincent, for setting the scene. So now is the moment where we want to hear from you and discuss with you. So we will now move to the first roundtable, and it's my pleasure to introduce the moderator of this roundtable, Gabriela Scarlatti, who will share this part of a session focusing on not business as usual. How do we move forward and foster new ideas? Not a small ask. Gabriela leads the Viral Evolution and Transmission Unit at the San Raffaele Scientific Institute in Milan, Italy, and served as Science Director of a global HIV vaccine enterprise from 2016 to 2017 in New York. Gabriela, the floor is yours. I mean, the floor is yours, actually, all of you, and all of you who are joining us online. Please do not be shy. Thank you.
Thank you so much. Nice to be here again. It's always a nice place to be and discuss science, and not only science. As Marlene said, it's a special year, but not only for the enterprise, it's a special year for HIV because it's 40 years since it was discovered. So it's 40 years that we don't have a vaccine, let's say. And that's obviously it's annoying us a lot, even if we made enormous progress in many aspects. And we heard it from both the fantastic presentations that we had here just today by Vincent and Sheila. One of the first things that I don't know if you had the pleasure to read it on the 20 years of the Global HIV Vaccine Enterprise. When I received it, I thought it was very well done. And I remember most of it from the past. And indeed, if you look at it, it's a kind of escalation that went from a very G8 endorsed through different phases of it, where we had collaborations, we had programs, but we need to get back to some G20, G something, and get people again, the politicians and globally recognize that a vaccine is still needed. And we heard that very much. Vincent, he really said it. It's a burden of disease is there, but it's not anymore a pandemic as we lived it 40 years ago. It is throughout the countries, everywhere in the world. But as he said, it's 60% are in sub-Saharan Africa, the new cases, and it's young women and girls. So we have to think about that very much. It's when we start thinking about vaccines and how we want them really to be developed and where we want them to be, where to be brought and where they have to be really applied. They have to, my opinion is that we have to vaccinate everybody, but all over the world, but certainly certain areas are heavily affected. And what Vincent also told us that he gave it immediately for granted. He said there is a need for treatment. That means that prevention doesn't work just now as it should, and that's clear, right? And also Sheila told us that the variants, we have to consider the variants that are there. It's not only B clades. Most of our vaccines are kind of very, very narrow oriented, I would say. I don't know, am I the only one thinking that? And but we have really to find new ways to approach that. So said that, and I think the other message that came through, and then we start, I close my introduction, is both of them told us that we need to be speedy. We need to have fast platforms. We need to find new ways, and that's also what we were asked to think out of the box today, and especially we need really new approaches, but new approaches that could be new vaccines. Possibly we have to start from scratch again. Oh, I hope not. But the other is how to really do new trials also. And both Sheila and Vincent, they pointed to experimental medical trials. We have to really go fast and be able to test many products in a fast way. But is it only products, or is it also other components of the vaccines that we need to test together with those? Because like EV 2020 did, I think, remind me, 9 clinical trials, experimental medical trials, testing one after the other different proteins, and we got very good results. But we didn't get a new product. We got a lot of interesting data, but still we have to go on and do a lot of new formulations, new approaches. And said that, I think that one of the, when we think that now it's 40 years, and while everybody always comments, yeah, but COVID did it very fast, right? It's very banal, how do you say? in English. Yes, mundane. But indeed, what was it that in COVID-19 made it such a difference? Was it the science? Did they take science from us, from the past? Or was it policy? What was it? Nothing.
It was a lot of things, but I think one critical difference is the lack of viral diversity in COVID compared to HIV.
Only?
Not only, but that's a big one.
Oh, nice.
Louder.
Yeah, sorry. I think we cannot compare two viruses. They are different viruses. One is respiratory. Most people have self-limiting infection. So that tells the immune correlated protection right there. HIV is an integrated virus. You cannot compare. So I think that's a not fair comparison. But on the other hand, the society response, we can compare. So the good example is society, the whole world, every country, every president, chairman, they all devote billion dollars, no matter what political system. I think that's the lesson we should learn. Also, you see from the Western country, what are the companies succeeded in COVID vaccine? Not big four vaccine pharma, two small biotech, Moderna and BioNTech. They almost went to bankruptcy just before the COVID. See, that's a lesson. We should have such fundamental seed in our society, and they are ready technically, supported by CEPI and other things. So that's why I think a lesson we should learn. That's why recently, Roger knows that, I have been promoting the idea of a biotech approach for HIV research. HIV vaccines so far are funded by NIH, by Gates, by many other big organizations. We treat it as a public sector responsibility, which should be the case. But at the same time, I think we should mobilize, let's stimulate the society as a biotech, the return, the charity, the greedy of investment. Every approach should be used. Otherwise, I think this job will be very hard. So I think Moderna, they started as a financial company. They went to IPO with no data, no public, no product at all. So this is the lesson we should learn. So I really thank Enterprise recently start this HIV vaccine partnership. I don't see the name here. I hope it's still existing. I think that's a good thing to promote, to mobilize, and to have more people to invest, to join our risky endeavor. I think that's the thing we need to have. That's one comparison I will make for this too, even though there are a lot of scientific and other things difference.
I think there are other comments also here around. You see?
Just a quick note, if you could just, whenever you're speaking, just introduce yourself briefly for our colleagues online and for those who will then watch the recording and listen, that would be great. Thank you very much.
Shan-Lu, sorry, is a long-lasting HIV researcher.
I keep hearing the traditional vaccine approach being labeled as bad thing, which I strongly disagree. A vaccine is a vaccine. There's no traditional allergy. Sorry. Still you might have to issue. See, it's not easy. Okay, I'm Shan-Lu. I'm a professor from University of Massachusetts Medical School. I've been an HIV researcher as far as I can remember, 30 years or so. I started from DNA vaccine. You can say that's very novel, but in my mind, I always think vaccine still today is an empirical science. If we don't think that way, we will fail. Tell you how many vaccine really the COVID, right? mRNA. It's empirical. People don't know, just gambled and pushed, succeeded. So how do you know? So scientific, I can tell you in literature, before COVID, mRNA vaccine as DNA vaccine was labeled as not very practical.
It was not, yes.
right?
it had
So
made-
did we forget it so far?
Yeah.
We all think everyone's so smart. We know mRNA vaccine will work. So learn the lesson. We cannot predict. We can only work. So that's why I retired from university, become an
Yeah.
emeritus professor. I'm joining the world, the only company in the world at this time, works the HIV vaccine, devoted to HIV vaccine development.
There are
That's
also
our mission.
other, thank you very much, Sean. There are also others that I saw him, I saw Tom shaking the head.
Well, maybe I should start by saying that I think the overall direction of the field and travel is good. We're getting closer to vaccine that we have ever been. It might still be far away, but the overall direction for T cells and also for antibody is good. I disagree with the previous speaker saying that small companies are the success. Small companies are success only if they join with AstraZeneca or Pfizer, and AstraZeneca invest 50 million into the development at risk without knowing whether this is going to be working or not. And of course, the main difference between HIV and COVID is that COVID was easy. If HIV was as easy as COVID, we would have vaccine 40 years ago, sorry.
No one was speaking about RNA 40 years ago, but so when we had very few variants.
Yeah, RNA is nice, but it would be nice, and we are all waiting for seeing the success of RNA for other vaccines and approaches for other diseases. And I think this is still yet to come. So
Yeah.
it's not a miraculous solution to all vaccines.
And certainly it doesn't look like that the normal mRNA is the way forward for an effective HIV vaccine as it is just now. Any other comments? Here you go.
Yeah. My name is Gus Cairns. I'm not a scientist. I always have to sort of tell you who I am to overcome slight imposter syndrome still now. I write for AIDS Map. I've been an HIV activist and also an interpreter of HIV science to the community, I suppose you'd say, since my own life was saved with antiretrovirals in 1998. And I followed the tail of the HIV vaccine for the last 20 years. I remember GHVE being founded. And I also became particularly interested in PrEP and was actually got involved in a couple of studies and things like that. Right. So I'm really interested in how this feeds through to the public perception of vaccines and of an HIV vaccine. And I want us not to make some of the same mistakes we made with COVID. The development of the COVID vaccine was amazing, you know, for a layperson like me to have it done so quickly. But I also think that there were several mistakes along the way. One was that it wasn't sufficiently well communicated to the public that this was a vaccine that could only be trialed in terms of its ability to stop illness and death. So that's fed into, I think, a public skepticism about vaccines, because people are saying, well, I still caught the Omicron variant and I had a booster. How come it didn't stop me getting it? And of course, it is effective against infection, but only partially so. And also, we don't want to stop at an HIV vaccine that's partially effective. We may get that will almost certainly have to be a step along the way. But I think in terms of what we give to the public, and with Vincent's list of what is desirable in an HIV vaccine in mind, I don't think we should stop at half measures. We need to have the full product. And that's partially because twice yearly injectable PrEP is imminent in terms of its licensing, although it may take longer to afford it. But by the time we have an HIV vaccine, we will probably have medications that prevent progression to AIDS in most people, or that largely prevent infection, that won't feel very functionally different from a vaccine you have to take once or twice a year. So we want something that is lifetime. It's something maybe you can give to children before they become sexually active. And so we don't need we mustn't stop at halfway there. And there are, of course, a couple of other things that we need to remember from COVID. One is cost and equity. There are still 7 million people a year dying of COVID, mainly in the lower income world, who haven't got the vaccine. And also, we must remember that, yes, 60% of people with HIV are still catching it in Africa, but that number is going down. Africa is, in its own terms, a success story. We actually sit in the WHO region, which is one of the two where HIV infections have not gone down. And one of the reasons for that is because of the stigma against HIV, which prevents people getting things like PrEP and antiretrovirals that render them non-infectious. And we only have to remember that, do you remember the hoo-ha about the HPV vaccine? Fantastically effective vaccine. Stops women getting cervical cancer. I think people are now convinced of it's right. But there was a terrific fuss about, you're giving this to school kids. Let's avoid that as well. Yeah. That's all I want to say right now. Thanks.
Thank you very much. It's, I would say that you brought up a few aspects, and one is, do we need sterilizing immunity for HIV? And that's one, came throughout several times. And PrEP is true, it's coming up. 2 shots should be sufficient, but I make always the example of mother-to-child transmission. 1994, we knew how to prevent it. It didn't work for many, many years all over the world, and it still doesn't work. So it takes time before you can use drugs to prevent really transmission. But what you said is, it brings us to one of the question that is, what shall vaccine research, R&D really focus on nowadays? Which are the aspects that we should really focus on? Oops, I have here someone. Oh, we have Jean-Daniel. Thank you for being here with us.
I think Martin raised his hand.
Hi. And Jean-Daniel is that he thinks that it's probably more relevant to compare malaria and HIV vaccines. Malaria vaccines and HIV vaccines, I mean, yeah. Do you want to say something about that?
No, just to mention that there are 2 complex pathogens, and we know that the immune response against these pathogens are quite difficult to obtain. However, at the end, we have now 1 or even 2 vaccines against malaria that are not 100% effective, but are quite important in the field. And for many years, we thought that it would not be possible to have a vaccine against malaria. So I think rather than COVID-19, which is completely new pathogens, and so the impact globally is completely different, we may focus more on a kind of comparison with a malaria vaccine. The other point I want to come back to is one you raised, Gabriela, at the beginning during your introduction, is who should we vaccinate? And I think currently we don't have enough clinical trials in children. We know that, for instance, it's easier to induce broadly neutralizing antibody in children than in adults. And I don't know who we are going to vaccinate if we have an effective vaccine, but maybe it will be children. So if we have poor, not so good immunogen, we may try them in children, because even if we have a poor protection, a very low protection, around 30 or 40%, could be sufficient. And combined with other tools, we may then have at least the first interesting vaccine.
Thank you. You know how much I love children. We are running just now PEDMAP trial in South Africa with BNAPs, but to prevent mother-to-child transmission through breastfeeding. So I agree very much that children are different, but unfortunately we have to do first adult trial before we can go into children. And thank you, but it's true, we have to think about that population as well as a different way of having a different immune response, especially when they're neonates and in the first few years of life. That's very important. I think Tom is running a vaccine trial in children. I see hands rising all over. That's nice. I like it. Go ahead.
Hi, this is Johan Wekemans, working at IAVI. And I sort of like the way you propose to identify two important questions. And one was, seems to be around defining the public health targets in front of us for HIV vaccine development. That narrative has to be renewed in view of the upcoming new platforms that will play a role in prevention. We've heard about long-acting PrEP, potentially monoclonal antibodies will play a role, but so clearly that narrative needs renewal, and there isn't going to be one ultimate solution for all. I think it's really important to reiterate that people need choice, and that appropriate technologies will differ according to the environment and the epidemiological setting. And so I think that narrative is important. And I think keeping, long-term goal to do with elimination as a public health target is really important. And I also think that around the notion of having a long-term target for immune vaccine-based approaches to cure could be of interest, that is for the long-term targets. This being said, I also think it's really important to be very realistic in the message and have a message of truth about where we are in the program. And maybe it's not all or nothing in terms of vaccine development being empirical versus evidence-based. And maybe we are moving to a stage where vaccine development can become less empirical and more evidence-based. And so on this R&D pathway, I think your second question was extremely important as well, which is what are the targets on the R&D pathways? Because I think there are targets that are really worth highlighting. And that includes generation of BNAPs, first in stringent animal models, and then in humans. And we actually know the pathway to generation of BNAPs. It's been pretty well characterized. And so actually just showing that we walk that pathway is of interest. And that's what we're trying to do at IAVI with the germline targeting vaccine program. I also think that we should work on the R&D targets for T-cell vaccine approaches. And one of interest is obviously to investigate viral control under treatment interruption studies. Maybe there are others as well. I mean, we can, in the lab, work on strain diversity, but maybe it would be useful to further define the appropriate clinical and laboratory targets for T-cell vaccines.
Thank you for this. Before you, Christian, we have the BNAP hero. Barton Haynes has raised his hand before you. So, Bart, are you online?
Hey, thank you very much. I really appreciate it.
Hearing you again.
It's nice to at least be here virtually. I'm sorry I couldn't be there in person. What a wonderful discussion. I just had several points to make. With regard to HIV and COVID, one, I think it's important to look at the biology of neutralizing antibodies. The body loves to make, and I think Shanlu mentioned this, that it was easy because the body loves to make these kinds of antibodies for COVID, and the body does not favor making broad neutralizing antibodies for lots of reasons, many of which were talked about in the excellent talk just we've had. And so HIV evolved to escape neutralizing antibody production, trying to overcome with the strategies of germline targeting and B lineage design and so forth. But the bar is very high, as also has been pointed out, because HIV is integrating. And yes, we do need to prevent infection. The CMV T-cell vaccine allows the first round of infection to occur, but cleans it up at that point, and in effect prevents persistent infection. For BNABs, they're going to have to completely prevent transmission events, and it's an incredibly high bar. And so we have had, and so with regard to answering your question, Gabriella, where do we go from here? I think we need to learn the rules of how to induce, basically how to induce broad neutralizing antibodies by engineering the B-cell arm of the immune system. And so we need to learn the rules of how do you manipulate the B-cell arm of the immune system to do what it normally is disfavored to do. And part of those rules are, we've figured out that you need to have an affinity gradient between the immunogens, that they need to have ever increasing affinity for the sequences what we're trying to induce. And also that broad neutralizing antibodies, because of the long journey they have to go to get to where they can broadly neutralize, they accumulate a huge number of very rare mutations, which are really quite bar for us to overcome as vaccinologists in trying to induce these in a short period of time. And then finally, Tom Hankey has spoken about T-cell immunogens and has excellent trials in place, and we're collaborating with Tom on combining his and Betty Corber's T-cell immunogens with our B-cell immunogens, because the first speaker correctly noted that we need both T and B-cell arms of the immune system to work. And finally, point I would make is that we do have an HV10 trial 133 with a paper submitted. It's also online in medRxiv, where we have gotten to heterologous neutralization with a liposome formulated GP41 epitope, and actually gotten in humans these improbable mutations in a broad neutralizing lineages. And so I think the journey is going to be long to get to where we want to go, but we're much, much further along than when many of us started, as Shanlu said, 30-something years ago. So I'm personally very optimistic about where we are in the field. Thank you for allowing me to talk.
Thank you, Bart. If I can ask you, what would you change now to be faster and more BNAB oriented?
Well, Sheila's talk
would be successful, certainly successful.
Yes, well, Sheila's talk was spot on with regard to, okay, the hard work in getting mRNA technology going. And Sheila, I just wrote the Pontiano while I was listening to your talk and said, we'll be happy to help in any way, because we've been struggling with this for now 8 years to get mRNA technology going and have it going. We believe that to iterate mRNA technology is the fastest way to go, and I applaud you for what you're doing in Uganda. Whether it's going to be the final vaccine platform remains to be seen because of reactogenicity and whether it induces long-lived plasma cells, which are going to be really critical for the final vaccine, et cetera. I don't know. But it's certainly to iterate. We're making both proteins and comparing them with mRNAs as we go forward. But as you pointed out, it takes years to make proteins and months to make mRNAs. So mRNAs are one way today to iterate, and we'll see how it does with regard to side effects and allergies and things like that. Secondly, I think what the HV10 is doing and going to the FDA and the Division of AIDS is doing this and trying to speed up the carrying out of the discovery trials, both in the United States and then working in countries outside the United States to speed up discovery trials is really very, very important. And the sooner we can get to a common platform, or at least a platform that is starting to succeed, then that should accelerate the discovery trials from the regulatory point of view. And third is what we're trying to do is to speed up the release of our products and actually the manufacture and release of our products as quickly as we can. So I think, and I know all of those involved around the world in clinical trials is trying to speed up the design and of the concepts and the writing of the clinical trials protocol, which traditionally takes several years. But I think that's being sped up as well. So in all those areas, I think we can make progress in speeding things, Gabrielle.
Thank you so much. Indeed, there is still a way to go with RNA platforms and to induce BNAbs, and certainly also different RNA platforms that are developed to have mature VLPs or different types of RNA self-replicating that we heard before. There is a long way to go, and you mentioned the discovery or the experimental medical trials. Those are possibly the way really to go fast and get results at this very fast speed. Obviously, that means we have to have also financial support for that and the population to test all this. Well,
Gabrielle, if I could just make one more comment, just to follow up on what you said, the key is the design of the immunogens, not so much the platform. And secondly, for the discovery trials, to put in a plug for the discovery trials, we know so much about what protective responses are at the T cell side and on the B cell side, that once we see what the immune responses are that are generated and how high level they are, I think we'll have a pretty good ability to predict the results of a phase 3 trial. So I think the discovery trials are fundamental to solving this particular problem.
That will make the big difference, because until now our prediction was not very successful. Hopefully in the future we will be much, much better. Thank you so much. I have Christian that had raised his hand since a while.
Okay, I'm Christian Brender, Barcelona. It goes back to your question, but it really from before, whether we need sterilizing immunity, but it goes really back into what just Bart says on the design. I do think with HIV, we want to have sterilizing immunity. Otherwise, it means we have breakthrough infection. That means reservoir. That means reactivation. If we allow for partially effective vaccine, then it needs to be judged on its ability to achieve a reservoir size that we all feel happy with and safe. No, but going to the how to get there, that was your other point. And Bart just said, we kind of know what we need to induce. We know the responses that are protective, and I fully agree on that. How we try to induce those, I think that's where we may go apart. Many of the, at least on the T cell side approaches, they try to learn from the virus and impose that onto the immune system. I would say we have it maybe easier if we try to understand the immune system and then to try to learn from that what to induce. And as an example on that, or a parallel, in the neutralizing antibody world, people have been identified with neutralizing activity, B cells isolated, neutralizing antibodies generated, monoclonals given passively, fantastically effective. Well, that's looking at the nature of the infection and learning for the therapy. And in a possibly shameless attempt of self-promotion, this is what Bea did in Barcelona and our lab when we looked at the infected people to build a T cell immunogen. We looked at 1,000 people asking what their responses are. And those responses have been induced with a platform that is more or less effective, gives certain rise, and has led to the best therapeutic use and induction of immune responses. And unfortunately, it's not popping up anywhere. And that's my personal frustration. But I think it goes back to exactly what you asked. We need to change what has not worked over the last 40 years. And I know that I'm the second generation behind that generation who is on it for 40 years, and that may disqualify me from talking, or exactly not. Plus, I'm not that much younger anyways. Exactly. But we cannot just keep on reinventing the wheel over the same things and dare to break loose and actually give credit that is not out of the establishment for things that come out of there. That's my concern, that we are still caught in keep on going the same ways that we have set the direction 30 years ago.
Yep, and we have to change that. How do we change it? That's the point. How do you go out of the easy way? You continue, go. Because it's easy to continue on that, right? Even if you have failures, but then you adjust. And
I'm looking to you for changing it, Gabriele.
No, I'm going to retire soon. Joking. Don't think so. Yes, I don't know your name. I apologize. Please.
Hi, my name is Daisy Oya, and
Hi.
I am with AVAC.
Oh.
Yeah,
Now
so
I can
this has been really interesting. It's clear that we need both the T cell and the BNAB response in order to have a successful vaccine, if I was following correctly. And my question is, how are we going to combine what is being tested? If the ERV vaccine is successful with the germline targeting to induce, BNABs, and then perhaps Tom Hankey's T-cell vaccine is successful, what is needed? And I was thinking that now is the time to think about it. Are we going to combine them into one vaccine, or is that is my big question to also Sheila, in terms of the way forward. Do we need a common platform, or are these vaccines, can they just be put together in a vial and then we make that? In terms of who to vaccinate, I think the COVID experience really hurt vaccine confidence, especially in our part of the world. I'm from Kenya, from Sub-Saharan Africa. And I think it's important for us to, as we pass these messages on to communities, to really try and build vaccine confidence, because we might end up with a vaccine that, we are in the post-truth world now, right? And a lot of times, especially the mRNA platform, there was a lot of rumors, and there still are, around vaccines. So in the past, we've been in the vaccine field for a long time, and we could go and say, everybody, Africa needs an HIV vaccine. And of course, that's the message, and it still is true right now. But now it's like, it's treated with a lot more suspicion than before. So I think the vaccine confidence arena cannot be forgotten, much as we are developing this vaccine. And I think I was also very fascinated about the vaccinal effect of BNAB administration. And I was wondering if any of the researchers in the room can talk about what it means for the vaccine discovery field. Thank you so much.
Thank you. There was a workshop 2 weeks ago that was done by, did you follow on that? Me too, unfortunately. I don't ask Christian to answer on the vaccinal effect of antibodies, because we have always been discussing he's pro and I'm against. And then sometimes it changes.
Not lately, guys. But that one question about combining, I think that's a comment I wanted to make to the breakthrough infections, especially there. If we were to allow for it, we need to have something that controls reservoir. And I think their T cells will need to play a role in it. Some people think also antibody will play a role to that as well. But combining it, Gabriela alluded to it, to the small experimental clinical trial that in EV, there were almost 10 or so. And I think that's the way to go. Small trials to eliminate the non-productive regimens or combinations. But first, we need to really understand whether there's actually an interference. There are monkey trials going on, and that's the animal model, I think, good enough for, but for efficacy, evidently not later on. But one can, I think, start exploring what candidate can go with what other candidate for inducing TMP immunogenicity in small trials. So we are talking with Robin Shattuck these days on a 10-arm study with 5 to 10 people in each arm, just to eliminate the least likely ones from that arsenal of different combinations. Because I think we are still in the darks of what will work and need to still empirically test it, but then also have to eliminate the ones that do not seem to give us good results or show an interference based on vectors or order of immunizations.
But there has to be the will to combine them, and you have to put different people together,
Absolutely.
because we all have our small garden where we cultivate our.
That's a discussion we had before the meeting with Roger already, you know, that people get out of the silos. But I think that has changed.
Yeah.
I think that's the benefit of the last few years, that people of TMP and innate immunity come together,
It's
really.
also, Sheila, you had something to say?
Yes, ideally, he has commented on the cocktail approach. And as Gabriela has highlighted, it's quite key to bring the different research groups together to harmonize and have people conduct collaborative research from an early stage. So it's quite key. And the way forward with the immense diversity is going to be a combined approach that is clear now. Yeah.
Thank you, Sheila. I think there is Tom next. I've seen you. No.
I just want to make 2 points, and I think they are best or most easier to demonstrate for me on T-cell vaccines. So we have to get lots of things right at the same time. It's not going to be only about immunogen. It's also its delivery, and the T cells will have to recognize the right sites. They will have to recognize multiple sites. They will have to proliferate. They will have to be robust enough not to be redirected to the non-protective sites. They will have to be at the right time, at the right place. And all these things have to be right. We miss one of these, goes wrong, and we lose the whole strategy. Is not going to work. The best immunogen in the world, if it's not presented correctly to the immune system, it's not going to induce protective responses. The same thing if the immunogen is wrong and we have lovely delivery RNA, again, it's not going to use protection. We have to have multiple things that have to be all right. And in particular for B cells, I want, for T cells, sorry, induction, I want to make one more point that I think we need heterologous prime boost, especially for vaccine vectors which don't replicate, in order to have sufficient expansion of these protective T cells to, because we have learned, and we keep repeating the same mistake, that if we deliver adenovirus 3 times, we're not inducing any responses anymore with the last boost. We've learned that from the STEP study, and guess what happens in Mosaico? We do the same. So heterologous prime boost for non-replicating vaccine vectors. Maybe replicating vaccine vectors, if they are safe, of course, they still a completely different immune responses, much more potent. And I stop here.
So we need small, fast trials to exclude what does not work or what we think does not work. We need combinations of different products. We need prime boost. I didn't hear anything about adjuvants, multiple administration, multiple antigens, but that comes with it. I think, suppose everything has to go with some antigen or almost adjuvants. And he's always pushing me to look at his. I don't know what. What is it?
Sorry, thank you. I have to say it myself. It's very interesting, and I think you did a great summary, Gabriela. But I was just wondering, we have different people with different ideas, and I got the feeling that it's not the first time that's happening. But is this happening often enough? Where are these conversations taking place? Should they take place more often? How can we ensure that this dialogue is not just a dialogue that happens 1 every 4 years, or just because there is a conference? Because what I see personally as a problem is that the exchange of ideas, the exchange of perspective, what's at the heart of science, you know, between the Plattist and the the others, is that this discussion are happening. So I think it's part of the what will move the science. Any view on that?
Sean?
I agree. I think we should have more discussion about different ideas. Now you go to most HIV vaccine meeting, actually, unfortunately, we don't have many. We're getting less and less. When 10 years ago, we have a dominant HIV vaccine meeting. Now HIV vaccine being wrapped into some other meetings together. We have less. And then go to other meetings still remaining, you only hear one dominant idea. It's a protein neutralizing antibody, which nothing is wrong with that. But on the other hand, I think we should have a more platform to allow different ideas to be presented. For example, during this session so far, from the speech introduction and the discussion, we hear two ideas. One is a protein neutralizing antibody, other is a T-cell response. But I did not hear or hear very little about the other the parameters. We learned the ADCC or V1, V2 antibody, because that's the only thing has been suggested as a correlated protection. We are dying, we are crying for correlated protection when we have it, but we are not just studying more. Of course, we have a scientific debate. Scientists always have a bias, what I like, what I don't like. But I think we should have an open discussion. Also, I may add that some data not fully published, but some published. For example, 2022, Journal of Infectious Disease published analysis by HIV10 group led by Peter Gibb. You can see that they still support the idea of a V1, V2 antibody is an immune correlated protection, even in a failed vaccine 702. And the other unpublished data, I think Zoe is here. I'm sure you will hear very soon when the data being published. Even in 705, the immune correlated protection, the V1, V2 still holds that. I think we should not throw out a potential good idea because the follow-up trials did not reproduce success. And also keep that in mind, 705, 706 was not designed based on RV144. They're completely different design. People just lump everything non-neutralizing antibody idea into one basket, which I do not agree. I think that science is beautiful. So many new questions we can ask. We should not just narrow put it away. So I fully agree with Roger. We should ask that. If we have, allow me, I can ask some simple scientific question. For example, do we know for the last 30 years, most HIV vaccine trials, especially go to efficacy, the envelope was modified and artificially designed, remodified, but they are not original virus sequence. Isn't that very shocking? We never tested as a control even. Also, the V1, V2, do you know V1, V2 was never in the famous crystal structure of HIV envelope, even to today. So how can we know? We know structural design, but the V1, V2 has suggested by efficacy trial saying there might be important. We don't have a structure. I don't mean that's a fault, but it's something showing our ignorance or language gap. We need to do a lot of research on that. Also, I think when Gabriel was the enterprise director, about 10 years, I don't remember when was that?
2016.
Yes, so very approaching 10 years. I remember you organized a workshop debate on the tier 1B neutralizing value. The overwhelming conclusion at the end was a tier 1B is a very useful parameter. And people supposed to write a summary. This never happened. Why? So I think this is all the scientific question. We don't have to do big idea. This is simple question. We are not following up. I think it's I will stop here.
So thank you very much. That means we have to, did you want to answer? No, because it was to you, in fact.
No, it was.
The important is not to work in silos and not to work only with your friends. It's usually much better, but sometimes that happens more often than we would like to. What did I want to say? No, my question is, what can the enterprise do in all this? Because the enterprise, as we said, it's 20 years, has been here. The vaccine enterprise has had different roles. What is the role now of the enterprise to really put together the people, share data, advocate for? What would you expect from the enterprise nowadays? Is that, am I allowed to ask that? Can we do without enterprise? Oh, I was not allowed to say that. We can't. Oh, don't say that. I really like.
No, that's.
No, I was looking at if there was some comment. No, the enterprise can do, because it has always been doing a lot of work. Oh, there is some.
Well, I'm not going to answer that question.
Oh, that's bad.
I'm Stacy from AVAC also. And it is something I think that this whole conversation is something that we are obviously thinking a lot about at AVAC, the future of HIV vaccine R&D, I think, especially in the wake of the Mosaico result earlier this year. I think everyone sort of recognized that was whatever analogy you want to use, but it was sort of I think the final disappointment, recognizing that there really needed to be kind of a shift in and I know this isn't exactly clearly the way it happened, but this sort of shifting into this idea of experimental medicine or discovery medicine, and I do wonder if what we need right now, as opposed to more meetings, I think getting people in a room and talking about things is helpful, but I do wonder if what we need is, and I really hesitate to say, but is sort of some sort of an articulation of a discovery medicine sort of strategy for the field. Johan, I think you said it well, like, what are the R&D targets? And we know that there are so many unanswered questions, but maybe sort of a better mapping of those R&D targets. And I'm like you, Gus, I'm speaking about this from a very non-scientific perspective, but sort of a mapping of those R&D targets, and then a mapping of the research players or the projects, and how they're all coming together to answer or reach those R&D targets, I think in a more systematic way than has been happening. Again, I think with this is sort of the shift that has happened kind of post-Mosaico. I'm thinking about, USAID. I think everyone knows AVAC works closely with USAID. USAID has this new project, Brilliant, that's starting up. And so just recognizing this is, the science is really, it seems like it's getting more complex and harder. I mean, these unanswered questions that we still have from the efficacy trials that have happened, and it's evolving so quickly. So maybe just, I think, a better, a more sort of comprehensive and articulated strategy. And then I think some sort of a mechanism to make sure we as a field are moving that strategy forward. So I don't know if the enterprise wants to do that, but an idea.
Thank you so much. And I think we have a few minutes left and we can go on. Oh. Okay, but I think I'm happy to, I know first, I apologize. I'm happy, I hope I can see you, Bill, because it's years that I don't see you any longer. And Bill has raised his hand, Bill Snow, the former director, or the last before this one. No, I don't know. Bill Snow, okay. Here you are, but only the picture. Okay. And Bill, you raised your hand. I would love to hear your comments. You had always fantastic ideas. I know. Can we hear Bill?
He's muted.
You're mute? No, we can't hear you. But meanwhile, you sort out Bill's mute problem. I think one of the questions that remain also is it too early to think about efficacy trials? Will we ever be able to run an efficacy trial, vaccine trial? Or do we have to think about, besides that we don't have yet a vaccine, but what are the hurdles just now that we have to face and think about that we might have to consider also in all our phase 1 and 2 trials and the design of our vaccine schedules? Do you think we will be able to do? How many years will it take to get the new vaccine in phase 2, and then we can run a phase 3 efficacy trial? How would you structure it? How would you design
If I
it?
allowed to say again, since I'm one of a developer.
Can you speak louder?
Poorly, I'm still working on HIV vaccine. And I kind of agree with Barhan said. The whole field, even after the Mosaic trial, is very depressing. But on the other hand—
That's not the first time that we get depressed.
That's right. On the other hand, as we mentioned earlier, the TB and malaria. Do you know the TB and malaria vaccine has a very major progress? So some people think HIV vaccine may also have a chance. Because so many years, we invest so much in immunology, biology, many other things. So the science is usually give you surprise. So I agree with Barhan's. He said that he has generated some heterologous neutralizing. I don't want to cite my own work. We also generated some CD4 binding site neutralizing monoclonal antibody from vaccinated people. I think progress will be made small steps. But on the other hand, being a developer, I'm also worried there's no mechanism for efficacy trial, no commitment from both the government and private sector. People are very depressed. They think that's not possible. So no mechanism. Even we have vaccine candidate, we are ready to there. I think this is two issues. We should move the pipeline to move forward, to continue the progress, the effort. But at the same time, no mechanism to me is very concerning.
Did you have, no, Tom and you?
Well, I just think that the realistic path is going to be that small cure trials are going to find out the vaccines that can make a difference for in clinic, and then with the cure results help, maybe we can gather some momentum for doing preventive trials. Unless, but of course we have to be aware that maybe the window for preventive efficacy trials is closing with the availability of antiretroviral treatment.
Especially long-acting antiretroviral treatment. But that's not going to be available all over the world that fast, I suppose. And finally, sorry, I kept you waiting.
No problem. No, I was thinking about clinical development pathways and being a little bit inspired by the experience of having closely worked on a malaria vaccine program that took 30 years and a COVID vaccine program that took 1 year. And I think there were differences that have been alluded to in terms of the funding available, in terms of the complexity of the science, but there was clearly a willingness to do things differently in the COVID regulatory space and clinical development avenues. And so I want to echo what has been said before about the need for seamless approaches to clinical testing to support the translational medicine research space, where we have clear ideas about scientific targets. And there is a need for a sense of urgency, but we shouldn't put, the downstream pathway the projection of the downstream pathway be an obstacle to what needs to be done upstream. And I think we do have a lot of things to do upstream in that research space. We need to make that research space agile and fast. And I think the enterprise and other bodies that put players together to really exchange on the science, and that has a lot of merit. And then preparing for the vision, I think we have to think long term, but it doesn't have to prevent acting, doing what it takes on the short term. And so working on the future of prevention trial, working on the future of access and public health needs is super important. But I think creating a platform for rapid iteration investigations of scientific concepts in the translation research space is super important.
We have a few minutes left, and I think there is no more comments. Margaret said that she agreed very much with you, Sean, and also with Bart's ideas, McCluskey. And I think we could make a round table and round of the table. And I would ask you, what would be your priority, or what you think is the priority we should pursue just now? One sentence, not two. Where shall we start? Tom, you are on the edge.
I take a deep breath in. Well, the one priority is not to be pessimistic. I don't want to hear at this table that there is post-mosaico and pre-mosaico, and we have to be. There is optimism. We've got the best T-cell vaccine results in cure ever. We have targeting successfully. One sentence. We are getting there. It's just hard and slow, but we are getting there, and we want to be positive. And certainly from this table, I don't want to see pessimism.
Very good. Go ahead. It's your turn.
I thought because I'm a laboratory, I'm exempted. Yeah, I think for me, it's just listening to what everyone is saying in the room. I think, yeah, the idea behind creating the enterprise was to strengthen, to push forward the collaboration, I think, among different stakeholders. And I think that's something that we need to keep pushing. I think, as you said, we don't need to work in silos or only working with your friends, but I think we need to continue strengthening the collaboration. I think that's the only way we're going to end up with an effective HIV vaccine.
Thank you.
Yeah, for me, I think we need to translate the message currently, because in the past we were talking about efficacy trials that were supposed to be working. I think now we need to really translate the message that the field has moved back to upstream, but that does not mean that we are losing hope. And so, and also just like work through the nuances, and there are many, especially in terms of community support, which we need quite desperately. Thank you.
I think I already said mine, but I'll just say it again. I mean, I think we need a much clearer sort of defining of the need for a vaccine. We need to go beyond sort of the world needs a vaccine, especially in the landscape of prevention and how it's going to evolve. And then, a clearer definition, I think, of the R&D targets and this sort of field-wide strategy.
One sentence for me is just try to be consistent with the approach, which well supported by the scientific and clinical result.
One sentence.
Give you a half sentence. Keep open-minded.
Very good. It's your turn.
I would say work together. Looks like there are still several gaps that need to be filled, but people are working aside. And so these type of meetings are really wonderful to put together all the ideas. And I think it should happen more often.
Christian?
I've spoken enough. Comment on Sean's only. Try to imagine that neutralization activity tells you about as much as interferon gamma release in an ELI spot. Be open-minded. Think about what actually your tests and analysis tell you about the functional correlate of protection and not just some correlate of what you observe with your data.
Please.
Yes, I was glad to hear Tom talk about learning from the cure trials, because certainly we are making progress with cure. Slow, but it's accelerating, I think, with more people actually being cured through stem cell transplants, but also more long-term viral suppression in studies with HEIs. And that, for me, there's more hints of what might be a true functional correlate in those trials than we may actually be getting from some of the analyses of the vaccine trials at the moment. So I think that's one way forward.
It's your turn, Marlene.
All right. I would say let's sustain collaborative approaches as we have in the room today. And I just want to take this opportunity also. We have more than 30 people online. So would also like to know, maybe we can't go over the 30 people and 20 people in the room, although which would be ideally the case, but would love also to hear more from our colleagues online about what they think is the way forward to foster new ideas and new approaches. But collaboration seems to be key when I'm hearing all of you, wherever we are working in the field. Thank you.
Thank you.
Yeah, so collaboration has been emphasized, and I think now looking at the data, as has been highlighted, it would be important to ensure that those critical gaps or missing areas are addressed before we move on to the next step. That will ensure that all areas and the baseline are covered before we move into the next area for vaccine development.
Thank you.
Thanks. And as a statistician in the room, I will say, I think innovative trial designs are going to be really critical in future. I hear a lot of talk about the need to move quickly and efficiently through, especially in the discovery medicine trials. And just a sobering, not negative thought is that there's a price to be paid for small trials. So I think we need to be really careful in dialing the numbers down too much. We don't get information for free. So there's a price to be paid there, but being clever about it and asking very clear questions that the data are capable of answering will be really critical.
Thank you. For me, I think it's echoed with the rest of my colleagues is how do we build ecosystems around the research and development pipeline? I think one of the things that I'm interested in that came out of the HVT full group meeting, and you can correct me if I'm wrong, but how do we infuse machine learning and artificial intelligence into the clinical research pipeline, and how do we make sure that community is up to speed with this new technology as products are being developed?
Thank you. I thought open-minded, open data, sharing of data. These are my big. If we share data, if we collaborate, we will get much faster where we want to go. And it's not easy to be on this side of the table when you heard already 15 of you. And then I have the pleasure to say what Margaret McCluskey has written here, if she doesn't want to say it herself. Margie?
Oh, sure, I'm happy to. Hi,
Good.
everybody. Good to be with you. Yeah, we used to have, so many things that have been said I agree with, not all, however. And one piece that I think is a struggle for me is that we have as a field so regularly gone one direction rather than diverse directions. That's been brought up.
Mm-hmm.
And I believe at this point, as being in the donor world for 17 years now, I realize how advantageous it would be if we coordinated better as donors and looked, especially now in this field, at who's doing broadly neutralizing antibody studies, why, and how do they complement each other. And I think donors may need to drive that conversation because for obvious reasons. So yeah, no, I think we're in a little bit of trouble. I agree with you, Tom, that we're probably closer than we've ever been, and we have more information than we've ever had, information we can count on. But I also think that we have some obstacles that are being ignored right now. And one of them is how we talk to one another and how we're coordinated, which is one of the reasons that the enterprise was born to begin with, is to decrease duplication and increase synergies. And I'm really not seeing that happen so much in the field right now. So I'll leave it at that. I'm sorry to not be more positive. I usually am very positive. But in this case, I think we need to be very realistic about the new platforms that are coming, about how long it will take for germline targeting to actually give us an answer, and that it's really the only approach on a product development path at the moment. And the lack of interrogation of what happened at V1, V2 in RV144. As my first comment, of course, I made the mistake V2, V3 because I'm multitasking, but the point is that that still is our only correlate, has been raised here, and it's not terribly sexy for most scientists to interrogate, and I think it's still an area that we need to look at. So I'll stop there.
Thank you very much. Nice seeing you again. And I think there is one more comment by Jean-Daniel. He says the priority is not to follow a single path, as Roger Tatou points out, to have much more regular discussions, and we should go on having more frequent meetings. Now it's your turn. No?
I'm just going to say, as an HIV prevention advocate, to make sure I've got the right hat, that for 40 years the virus has been evolving and has been one step ahead of us. What I would love to see is the field to evolve, catch up with the virus, and be one step ahead of the virus, so that we can finally control and manage, get rid of this epidemic.
I want to fuel the optimism and remind people that TB and malaria have been scourges for humanity for thousands and thousands of years. So let's not be desperate about 40 years in the desert. We have done a lot and we can do much more. So let's get out of the desert.
Thank you. Go ahead, Birgit.
Sure. Sure, Birgit from the International AIDS Society. My comment is probably better for the next session, but I won't be able to attend that one. I've been, thinking about also what Daisy was saying. But we need to have better messaging, and you've been talking about the kind of scientific message out there, and I'm not a scientist, so you need to do that. But I'm thinking about how do we talk to funders as well. And obviously there's the argument around the need and incidence and, get that under control. But I think there's also the argument of the excellence of science that is happening in HIV vaccine research. You all are doing top-notch science here, and it's not been for nothing over the last 40 years. Without all the HIV vaccine R&D, the networks on top of that, we still wouldn't have had the COVID vaccine as quickly. So it's not wasted investments. And I think we need to keep that argument in mind and reiterate that as well, particularly I think here in Europe, where we don't see governments put enough attention into that.
Thank you very much. We have other two. Go ahead.
Hi, everyone. I'm Zach from the European Health Management Association, and I'm very, very new to understanding HIV vaccine research. But what I will say to jump on the optimism train is that I see an immense strength of purpose and resilience in the room and in the discussions that you're having. And I think that's something to harness and hold on to moving forward.
Thanks, Zachary. My name is George Valiotis. I'm the Executive Director of the European Health Management Association. I work with Zachary, and what I would say is what I learned from the early HIV response is who got involved and how they got involved, and that diversity of people in the room was essential to what we did. What we know from our work in health management across Europe is that we are interested in lessening the burden on our healthcare systems. Every decision we make, especially for COVID, was about how do we lessen the burden on our healthcare systems. Prevention is not there. When prevention does enter, it enters because of, I think recently, of RSV. The RSV activism came from families, parents, people affected by RSV, people who didn't have RSV and who didn't want to have it. So what I would say in my thoughts are, I look forward to the advocacy session next, and I would say you have incredible skill and knowledge here, and you need to add to it and enrich it in order to be heard differently by other people. You need to add more people and more thinking to this room, I believe.
Thank you very much. It was a great discussion. Indeed, when I was the science director at the enterprise, I don't remember how many of those meetings we did very fast, a few hours only. And we managed always to have 100 people online because you could, the whole world, we had them in the middle of the day so that people from Australia could listen in to by midnight, others at early morning. But it was one way to get everybody online. And here we did the same. I mean, we have 20 people in the room, 25, another 30 that are sitting somewhere in the world around and attending and giving their opinion. And that's really fantastic. I think that's the way forward, opening up and discussing more and more and getting new ideas. So I leave the word to you, I think. You have to close this session.
Thank you, Gabriela. So I'd like just to thank all the speakers in the rooms and all of you who have been participating in this roundtable and those online who are joining with us for this session. We're going to have a short 10 minutes break. So please stay with us and we will resume very soon. Thank you.
For all of you, coffee is in the other room.
All right, we're going to resume now. Thank you again for rejoining. We're going to start with part 2, which is reinventing HIV vaccine advocacy. And it's my pleasure to introduce Mitchell Warren for a presentation on HIV Resource Tracker, an overview of current states of funding and trends. Mitchell Warren has been the executive director of AVAC since 2004. He leads the organization's overall program strategy and maintains strategic partnership with AIDS service organizations, research and development agencies, policymakers, funders, the media, and other health advocacy organizations. And additionally, Mitchell serves as governing council member of the International AIDS Society. Mitchell, the floor is yours.
Fantastic, Marlene. Thank you. Shall I share my screen? And can you hear me?
Yes, we can hear you well.
Great. And shall I share my screen?
No, the team will share your screen. You just have to let us know whenever you want to move slides.
Fantastic. You know, I don't even, maybe I'll just go slide. I can maybe move without any slides because much of what I'm about to describe has been pretty well articulated in the last couple of hours. And I'm sorry not to be with all of you in Geneva. I would love that. But I'm joining remotely instead. But so many wise words were said in the last couple of hours. And I do want to pick up on something, Tom, you said. I don't think I, or frankly, any of us could be in this room if we didn't have optimism. To be a vaccine advocate, to be an HIV vaccine researcher has as a precondition the need to be an optimist. So I come to you with optimism. I come to you with some forms of exhaustion and hopefully some attempts to recalibrate where we all need to go forward. And I'm going to try to use the resource tracking data to tell some stories about where the field has been, where it's going, and perhaps most importantly, what we all need to do to be sure that we get on track to deliver on a mission that I know we all share. So if you go to the next slide. And I did send an updated deck just a few minutes ago, and I don't know if this one is there, but go ahead and do the next one. Oh, it's not advancing for me. But what I was, there we go. Oh, good, you have the new slides. So happy anniversary. You know, when I was talking with the team last week, we talked a lot about the fact that it's 20 years. And I think Bart may have jumped off. I don't know if he's back. But I must say, I put this slide in because I realized in this list of authors since 2003, there are very few people left in the organizations that they were in at the time. I think really, except for Bart and Larry, we are looking at an entirely new cadre of leaders. These were mainly from funding agencies and research groups. I actually had this came out just before I joined AVAC, and I was working for Seth at IAVI, and Stacy and Daisy and a number of us kind of watched the enterprise emerge back then. And I'm going to come back to this closing paragraph, because much of what was said 20 years ago by this rather impressive group of authors actually is exactly what you all have said for the last couple of hours. And I don't say that in a sense that you all repeat and we all repeat ourselves. I say it in a sense that what we said 20 years ago, what we committed to 20 years ago, is just as relevant, and I would even argue more urgent today. And we'll come back to that in the course of the next little while. So we go to the next slide. I was asked to provide primarily a sense of the funding landscape. And this is where I want to calibrate expectations. Yes, we need more money. And in fact, one of the early enterprise documents said that we needed to get to $1 billion a year to get to an HIV vaccine. I think we all now know 2 decades later that it's not a singular amount of money that's going to get us an HIV vaccine. If this were just about money, I believe we would have cracked this. Yes, we need more money, but this is an incredibly well-funded enterprise, lowercase e, in the sense of that research. And this is based on now 20 years of data tracking that we have done in partnership with IAVI and UNAIDS, and having created the HIV Research and Development Resource Tracking Working Group. And now we have data going back 20 years. Resource tracking that we do for HIV prevention, much like GFinder, which does tracking across the full global health architecture, are imperfect sciences. There's a lot of data that we don't get, particularly from industry partners. But one of the great things about it is that over the course of 20 years, we can begin to see trends. And so every year the data gets richer, even though it remains somewhat imperfect. And so I'm going to be presenting most recent data from 2021. The 2022 data are just being finalized, and give you a sense of what I think it might mean for the field. So if you go to the next slide. This is overall across all of HIV prevention. And it's interesting because I just was in Paris a couple of weeks ago for the Union TB conference, and it really is remarkable. And I think Johan and others talked about the importance of thinking about TB, and it really is a remarkable moment. The TB vaccine field has less money than most of the colored numbers on this graphic. In fact, it is woefully underfunded for a disease that, as Johan said, has been around for many more decades than HIV. And yet one of the things that many people were saying around the halls of the Paris Conference Center, which I will say was the host of an IAS conference, and I remembered that we should never have conferences in that conference center, just as a little memo to all of us in conference planning. But one of the things that comes up so often in the TB world is right now, people saying, oh, we really wish we had more things in upstream. We have 6 products in late stage efficacy and a couple actually on tracks to licensure with pretty good phase 2B data to support them, but we've got nothing upstream. And they really see that as a disadvantaged pipeline. And I sit here in the HIV world in that meeting and thought, wow, what an embarrassment of riches to have so many shots on goal, so to speak, at this point. So it's all relative, and I just want to put that out there at the outset. And what's relative here is that for all the talk of treatment as prevention, for PrEP, for microbicides, which still are urgently needed, they are fractionally invested in compared to HIV vaccines. So you can see that this amounts on the year about $1.2 billion, of which 800 million of it is for HIV vaccines. And we have to remember as advocates how that might play out in the HIV world, but also in the larger global health world. If we say we don't have enough money, imagine what a TB vaccine advocate or a malaria vaccine advocate or a PrEP advocate might say. So we need to be sure that we understand our audiences as we use these data. If you go to the next slide. This gives you a sense of where the money's coming from. This is a public sector investment pool. And while we have seen changes over time, and obviously many of you will know that there were major corporate investments on the previous slide for Merck. And those disappeared when the STEP and Pambili trials were stopped early in great disappointment. And then we got a scale up in industry investments with J&J, and of course those are now coming to a close. Although through 2021, the data I'm presenting today, J&J's data are still fairly robust. That's going to change because right now we don't have large-scale pharmaceutical partners investing in vaccine R&D. It doesn't mean they're not engaged in it, but they're not investing their own resources. And we'll come back to that in a minute as well. But you can see the disproportionate nature. If it weren't for North America, primarily the United States, and if it weren't for South Africa, which I will highlight here, is a darker shade than other parts of the world, is one of the major investors in HIV vaccine R&D. You go to the next slide. This is vaccines specifically, and it has changed dramatically, obviously, since 2006, really. And I will note again, I should have put a little asterisk on this chart. 2004 is when or 2003 was when the enterprise was created, and it called for more resources. And we saw an increase then from 550 to almost 700 million, and that has continued. And then you see the dips after 2007. And again, if I map against this timeline, or if I map against this bar chart, the research results, of course, in 2007, that was when you saw Merck investing quite heavily in their adeno5 vector program. That disappeared after 2007, and you can see again in 2016, the uptick in industry again with J&J. But what's clear is that without, if you go back one slide, without the US government, and that's an aggregate of the US government, but you can see the NIH, the Military HIV Research Program, and USAID really driving this. And if you go to the next slide now, you can see the quote unquote league table of HIV investments. Do we need more money from Europe? Absolutely. Do we need more money from many other countries in the world? Absolutely. Do we need more philanthropy? Yes. But at the end of the day, and this is very consistent with the G-Finder data across all of global health, the NIH and the Gates Foundation drive R&D. That is not a huge surprise. We shouldn't despair. That is the reality across the entire global health landscape. The issue is, how do we find places where additional donors can make novel investments? And I think the Military Research Program and USAID, as well as the South African Medical Research Council, really demonstrate where they have found opportunities to invest that are additive. So it doesn't mean you have to spend $60 million a year to have impact. If you look at the EDCTP, of course, funding the current PrEP-Vac trial is a great example of a funder with much more modest resources still providing. But I think back to something Stacy said earlier, we need to develop as a field the clarity of the strategy, something Jose Esparza said at the very beginning of the enterprise. We need to develop that menu for investors to see where a $3 million investment might add value, because no one is going to compete with the Gates Foundation and the NIH. I don't think, I hope I'm wrong. We did see a big difference, I will say, in the TB world, where the Wellcome Trust was crowdsourced in with the Gates Foundation to fund a $550 million program in the latest M72 TB vaccine trial. So money, much like with COVID, can be found in the seat cushions of donors when there's a scientific rationale, when there's scientific promise. We go to the next slide. This gives you a sense of where this funding breaks down across research stage. And you can see, and this does change year on year, we expect that we will see in 2023 going forward, the clinical program may decrease. Now, obviously, there's going to be this explosion of discovery medicine studies, but those are a fraction of the cost of an efficacy trial. So in 2021, remember, the AMP trials were just winding down, which are included in this vaccine investment. Antibodies are included in the vaccine pot in the resource tracking. Imbokodo was still going on. It had just closed, but was still winding down with final visits. And Mosaico was still actually even enrolling in 2021. But you're going to see in 2023 and 2024, with PrEP-Vac ending, the last efficacy trial right now in the field, that I suspect the clinical piece of that pie will go down. That is not a bad thing, as long as we are investing again in the best basic and preclinical and early clinical development programs. Of course, I can't help but say that policy and advocacy can benefit from more resources. You've seen different versions of this from both Vincent and Sheila. Just gives you a sense that in the course of the timeline of those investments, the rise and mostly fall of efficacy trials with our two modest results, one in Thailand, and then of course the AMP studies, again, in this vaccine context, giving us hints, clues, ideas of new ways forward. But you can see that is now coming to a close now that Mosaico is done, and as I say, PrepVac will read out next year. If you go to the next slide. Additional context. I know already there's been conversation about where do vaccines fit? Where will a vaccine fit in the future for delivery? Where does a vaccine fit in the current trajectory? This shows you products that are currently in the HIV prevention pipeline that are nearest to market. And I'm not going to get into the details here. There is no vaccine or antibody in this slide. And I suspect there won't be until 26 or 2027 at the earliest. It doesn't mean we won't see efficacy trials, and obviously this issue of trial design is going to be critical, and I'm glad, Zoe, you brought that up. It's so critical. And yet, in terms of getting products to market, this is what the world looks like for the next 5 years. We shouldn't be depressed about that. We should actually be excited. This is a better opportunity to deliver prevention than ever before. But this is the context in which future trials are going to have to exist. And so we need to do this not as a way that says, oh, well, PrEP won't work. I actually think I know PrEP does work, and I think we can make it work better. It will not prevent the need for a vaccine. It does raise the bar, though, on both what a vaccine needs to look like compared to, for example, lenacapavir in the middle of the slide here. We'll get a result in 2025. Will a twice monthly injection work? And if it does, what does that mean for the target product profiles of future vaccines and antibodies? I have thoughts. I bet every one of you online and in the room has a thought, but none of us really know. But that's the conversation we need to be having. And the other conversation, again, is how will we study a future antibody or vaccine in this new dynamic context? You go to the next slide. This just is a wider view, again, showing the diversity of opportunity upstream, including the range of vaccines and also antibodies that Vincent and Sheila described, but in the context of a very dynamic space right now. Next slide. And this shows you what can happen when you have an easy pathogen. And you all have discussed this in the morning, just to give you a sense of how in the 2.5-year window, we actually had an incredible cascade of HIV prevention, vaccine, antibody, and injectable results in the same 3-year window that multiple vaccines for SARS-CoV-2 were discovered, developed, studied, emergency use authorized, then licensed, and rolled out, with all of the challenges that we've heard, and Daisy described it incredibly beautifully, the challenge we have, though, even in the context of those successes. But just again, context matters. And again, this is, I think Birgit had to leave, and I'm so glad she mentioned it. This just shows you that the return on investment is not, cannot, should not only be measured in a licensed product. What the HIV vaccine enterprise, capital E and lowercase e, have delivered in the last 20 years, beyond the fact that SARS-CoV-2 was easy, but everything else about developing those vaccines was made easier because of the people, because of the processes, because of the participatory practices that existed thanks to HIV vaccines. Without this enterprise, we would not have had COVID vaccines as quickly as we did. And we need to understand that as a return on the long time investment in HIV vaccines. And we also need to then look at what is the return on investment from COVID back into HIV. And I think Sheila described really nicely the potential of mRNA, a platform first studied, and I was so happy that when Drew Weissman was co-awarded the Nobel Prize, it was actually, it was reminding everybody that his first work in mRNA was because of HIV vaccine investments. That's a critical point. Well, what can we now get back? So what are some key takeaways in all of this? And I'm stating the obvious. We have to sustain the current level. 2022 data are not yet finalized, but we're not going to, I don't think we'll see much less than 2021. We'll probably be around that $800 million piece. So this is not a question of how much more money do we need, but how do we make sure that $800 million is spent to best effect? And again, I think that gets back to my colleague Stacey Hanna, who's earlier said this is the time to develop the specificity of the discovery agenda so that we know that $800 million is being spent well. How do we diversify investments and investors? And how do we find unique ways where smaller pots of money can add value? And perhaps, again, picking up on what you all mentioned earlier, what's the future look like for the target product, for the trial designs, for how we do it and who will pay for it? And then very importantly, I think it's when will industry come back? I think the honest truth is they'll come back when there's a scientific rationale to come back. Some of the biotechs are involved, certainly Vir and Moderna now working closely with IAVI. But how do we make sure that we get bigger pharma who will have the capacity to take products to scale if we ever are lucky enough? But at the same time, how do we make sure that the Modernas of the world do far better with equity and access with HIV vaccines than they did with COVID vaccines? So it's really great to get biotechs and pharma involved, but we have to make sure they do better than they've done with past technologies. And then finally, in the next slide, my hit list of 6 things that advocacy needs to focus on. None of this is going to surprise you because if you flip forward one more slide, I originally did this based on the lessons of COVID-19. But if you go back now, if you go one more slide forward, but harking back to the 20th anniversary of the enterprise, this is the concluding paragraph in that policy document from the August authorship that created the vaccine enterprise. And you can see that I have highlighted, and I had actually come up with this 6-point plan. We at AVAC came up with it many months ago. And when I was comparing it now this week to the 20th anniversary of the enterprise, how much history has both changed and how much it hasn't in terms of the work ahead. Many of the exact same issues identified by Bart and Larry and colleagues 20 years ago are what we need to be doing now with passion, with urgency, with optimism, because that's the only thing that is going to keep this enterprise alive. And I think if you go to the next slide, an article that a number of us wrote back in 2022 that, again, I think stands the test of time, and I encourage people to look at that. And then the next slide, just to give you a quick piece, we just this week launched From the Lab to the Jab, a way to help advocates and policymakers understand 4 interconnected aspects of how to move concepts from the laboratory to the jab to the actual vaccines. And we certainly encourage you to download those and use them in your work as advocates, as researchers, as policymakers. And with that, thank you and really look forward to the conversation. And thank you to all of our funders that make this work possible, and particularly to USAID and the Gates Foundation.
Thanks a lot, Mitchell. So we're going to move to the roundtable, and I hope it will be as vivid as it was for the first one. And we really want to hear from people in the room and people in the virtual room joining us online. And I'm very happy to introduce Roger Tatou as our moderator for this upcoming roundtable discussion. Roger Tatou is a global health consultant from France, and he's been working with the IS and the enterprise for a very long time now. So Roger, the floor is yours.
Thank you, Marlene, and thank you, Mitchell, for laying the ground so nicely and really helping me. There's so many things you said that I won't have to say, and you said them so much better than I could. We've lost a few scientists in the room here. People had to go. I know we have lost a few or so online, but although we are going to talk about advocacy, I really urge the scientists to stay online and to contribute, because good advocacy can only be built on good science, and scientists have a key role to play in building an efficient and an effective, I would say, using clinical trial word, advocacy. So as an introduction, I think it's fair to say, and it's broadly recognized, that the HIV vaccine R&D field is very much back to the bench, back to the discovery stage. We have had many failures, but we also have a significant pipeline of new promising product, vector approaches that we have heard of today. Nevertheless, and we also need to be honest with ourselves, the next HIV efficacy trial is at least 10 years plus from today. 10 years from now, the HIV prevention field will have moved. We may have, well, as Gus said earlier, twice yearly long-acting PrEP. And understanding what a vaccine will do in that context in 10 years from now, maybe 15, is very important. In parallel, global health has moved also. Global health priority has moved, have moved. Whether it's in high-income country or in low and in LMIC, the priority are changing. And it's clear also that, you know, HIV may not be as high as it was on the priority of Ministry of Health in many countries. So with the progress we have made with PrEP and long-acting PrEP, when I talk to people in the industry, when I talk to scientists and to advocate, is really how can we advocate efficiently, effectively for an HIV vaccine? How can we convince people that it's still needed? And to answer that question is really understanding, who do we need to talk to? What are we going to talk to these people? Who is going to talk to these people who make decisions, this decision maker that we don't always know? And how are we going to convey the message? So I think I would like to articulate this discussion around these 4 axes. And ready to start, I'm going to ask my good old friend Gus, who recently published three summaries, because he's doing a fantastic job at that, summaries of recent development with PrEP, showing that if both in the UK and France we're making great progress toward PrEP access, PrEP initiation, reducing number of infection, we're still not there, and we still won't be there in 2030, 7 years from now. So my first question will be, maybe the way to talk about vaccine is to talk about what we think should work, but in the real world doesn't work as well as we hope. So guys, PrEP's not working. Is that good enough to make, or not working as well as we want? Is that good enough point to make to support vaccine advocacy?
Oh dear.
Short answer. We want everybody to contribute.
Yeah, not simple. I mean, okay, slight anecdote. I remember when the results of the PROUD trial came out, which I was honoured to be co-chair of in 1985. I was talking to the guy then who was probably the HIV's top HIV consultant, Professor Brian Gazzard, and I remember him saying to me, "Oh, well, that's it then. You know, we've got HIV prevention sorted." What happened then was that even in the UK, we had to sue our own government in order to get PrEP approved. So, I mean, but I think that's an awfully long way down the line for an HIV vaccine, and the arguments need to be different. I think PrEP suffered from being seen as the gay men's approach to HIV prevention. And only now are we realizing that that may have laid a trap, as it were, for the usefulness and the access of PrEP to, for instance, women, especially in Africa, but also cisgender women everywhere else. And so I think we need to be aware, as I said in my when I talked previously, about the sort of the traps that may lie for us along the way in terms of stigma and simply sort of not being willing to address the issues getting in the way of the fair distribution and fair research funding into an HIV vaccine. For instance, what happens if it becomes clear that children are much better candidates for an HIV vaccine in terms of their immune response and also in terms of the age at which it needs to be applied than adults? Because then we will have a problem if we can't prove efficacy in adults first. Then we'll have to go straight to children. Now, that poses an ethical and a political dilemma. I'm not necessarily anticipating that will be the case. But I think we're a long way off that sort of thing yet. I think we're much more engaged with the what we've been talking about this afternoon about the different aspects of HIV prevention research, communicating with each other and acknowledging that a combination approach of different immune responses is likely to be needed. And the other thing I can never explain is why Europe doesn't get behind this so much. You know, I'm probably going to visit CERN tomorrow just out of curiosity. So that shows that Europe can do groundbreaking science at huge expense. Why aren't they behind an HIV vaccine? I don't know.
Thanks, Gus. So it's great because you just said the key word I wanted to hear, the groundbreaking science. We had a meeting here in June about European stakeholders, and we had about the same question. How do we convince people, the right people, still to be identified, that a vaccine is needed? And one of the things that was said first was that science is not going to be enough, that to have good science is not what's going to support the case for people to invest in more HIV vaccine research. It doesn't matter how good it is, people will still go back to, but we have ARV, we have Q equal Q, and that these are messages that are rightly promoted. And this is what advocates, and we have 3 in the room, which I will bring later in the conversation, are really pushing forward. But relying on the supply of drug for HIV prevention, is that really the way forward? We just have to look at the world around us, at the situation in Ukraine, in the Middle East, everywhere else. Can we just rely on, can we really rely on drug supply? And can we again, and that was the word of Jim Pickett recently in a call, can we rely, we can't put the onus on people living with HIV to take their pills so that Q equal Q works. So crafting the message around the need for HIV vaccine, and I'm going to give way, concluding that, one of the things we have seen with COVID and with recent development is the language as becoming extremely muddled. And when we talk about vaccine for prevention, but also vaccine for cure, that does not compute in many people's head. When we talk about PrEP and infused intervention being like a vaccine, this further complicates the situation. So I'll go back to my question. I'm going to open the floor. What message should we convey, and how do we convey it? Views. And again, if you're online, please raise your hand. Someone could tell me if someone raised their hand. What argument can we use beyond the science? And Tom, you already some of the answer because you were at this meeting, but you can jump in.
Well, to me, the argument is very simple. If you invest now, you're going to save much more in the future. And every politician and economist have to understand that. The second sort of point is that what I get frustrated with in this field and in publications about vaccine trials is complete lack of description of the vaccine. We agonizing about vaccines are failing and this is a failure. Does any of these articles actually look, this is the vaccine, these are the components, these are the immunogens that we're trying to, that we are delivering. These are the vectors that we are using. Look at this. If I want to induce protective T cells, what would I do? Can I improve this vaccine in any way? And you can very quickly find out that the delivery and the immunogens are actually a bit old, which might be with the time that it takes to actually get to this stage from the vaccine design. Same for antibodies induction. Look at the immunogens that we use in failed vaccine trials. Are they the state of the art, how we want to induce broadly neutralizing antibodies? Or is this something from 10 years ago that we were using and so we were progressing? So that is my frustration. We're talking about vaccine trials and we completely ignore to discuss the vaccines that are being tested. And finally, the third point is that the success of EPI of childhood vaccines is within the first half a year of life. That's when the immune response is ready to respond strongly to new immunogens. And that's where the opportunity is, but it is, of course, difficult because exactly of the young age. Stop.
Thank you. And if there are people in the room who want to comment or on phone.
Just a quick comment. Yeah, I wasn't just talking about immune response. I was also with my brain thinking about the HPV vaccine, about giving it to adolescents before the age of 6 as well, that issue.
So, Tom, you talk about a vaccine and describing the vaccine. So I'm going to jump on that and taking the same approach. But we heard today, I think at least 3 definitions of what a vaccine is. We had the vaccine that would provide sterilizing immunity, with a vaccine that would reduce viral load and improve long-term outcome. We had a vaccine about monoclonal antibodies. I'm going to look at my colleague from advocacy. When you talk to people, how much do they understand of this, and how important it is they understand? Because maybe it's not important, but I'm going to ask Maureen then Daisy. Do people understand all this, I would say, gobbledygook from scientists?
Well, so earlier this, I think I should say 2 months ago in Malawi, we just launched the Path to Scale, which is injectable PrEP implementation of science study. And as we're doing the launch, of course, it was a slip of the tongue from the Minister of Health. She said something like, the Kabele was a vaccine, and that has gone viral. So now there's a lot of demand for Kabere. Of course, unfortunately, as you know, with global supply, as a country, we're also trying to limit the number of people that can be initiated because we don't have enough in the pipeline. And the reason why I'm saying that, it's because there's a lot of work that we always have to do as advocates to explain the science, and it's not always easy. I think for me, just listening, yes, to the different type of vaccines, as you're saying, I was like, how can I break this down to a lay person so that they do understand the different, vaccines, the mechanisms? As I was listening to the presentation from Sheila and from Dr. Vincent, I was like, how can I break down this science? So it's not always easy, but I think where we're going right now, we just need to find a way, so that, yes, I think we are talking about the need to engage funders so that they can continue to put more resources, invest in, as Mitchell said, in novel strategies. But I think we also we don't need to leave the communities behind. I think from what Daisy said, where we saw with COVID, the vaccine confidence, I think it just demonstrated the importance of making sure that even with the most complex strategies, candidates that we can think of, I think we still need to find a way of making sure that communities do understand, because when we have efficacious products, we have products that have shown to work in clinical trials, now we need to give them to the people. That's where, communities still need to understand, they still need to understand how will that product work in their body. So as much as, I always say, yeah, nothing is too scientific for a community member, right? I think we still need to put in more effort to make sure that, they do understand. I know I didn't respond to your question, but it's something that we grapple with. And it's not always easy to break down the science, but it's always important, especially once those products are available and we need them to adhere, to use those products, yeah.
You're right, it's important community to understand. And, Daisy, what do you think? Are we being clear in our advocacy message? Do people understand what we are talking about, and do the right people understand what we are talking about?
Yeah, those are great questions, Roger. And I think the current landscape, is a completely different landscape. It's not like when we were dealing with the STEP trials and these other efficacy trials, because I think there is a lot of people now who don't even know about, AIDS. Maybe they've heard of HIV, but I think there's a generation now that have not felt or are not aware of like the impacts of HIV and AIDS, as, for instance, my generation, because we've been in this since maybe 15, 20 years ago. And I think this is all, of course, thanks to treatment and prevention and so on. And I think there's a lot of distraction. So what I'm trying to say is that the ground is not very much ready to receive our HIV vaccine message because there are very many conflicting priorities. There's crises, there's, as you were saying, wars and climate change, climate crisis, there's hunger, there's so many other competing priorities. So even the ground for us to be telling people about the HIV vaccine message is very, very unprepared, I think. And I think, there's also the issue that some quarters, some very well-respected quarters are saying, oh no, we can just end HIV using current prevention options. And, we've had that message, including from people like UNAIDS and so on. I think it's more nuanced, but, that's the key message that comes out when it's, put in a title. So the other thing is now the research is at a very early stage. And so I think we need to really nuance that message, find ways to communicate that even those clinical trials where we were doing efficacy for work came from basic science and so on. And I think that is now, I think, the challenge for advocacy in order to get the support that we need from communities. There's always been a question of at what point are you going to start involving communities in this? Is it very basic science? Because I think maybe 90% of the research that happens in, preclinical and so on, and even phase 1, phase 0, will never see the light of day. So at what point are you going to be starting to communicate? So I think I'll stop there. So I'll let Stacey say something. Thank you.
Yeah, Stacey, go ahead, please.
No, I mean, I think Maureen and Daisy have said a lot of really important things. You asked this question about what does community understand about vaccines or an HIV vaccine? And I mean, I think for me, one of the real challenges there is this use of the term community. I think we oftentimes just sort of use community really broadly and think that we just need this sort of huge community movement, for X, Y, or Z. And I don't think that that's where we are with HIV vaccine advocacy right now. So I think it's a matter of defining the community, and then what we need from them. Because I think when you go to the community and try to sort of increase their research literacy or their vaccine literacy, they always say, well, what do you need from me? So I think it's a matter of us, sitting here and really thinking about the challenges that we're trying to work through in terms of the future of the science and actually, getting us to a successful vaccine and identifying, what are the needs. So if you look at it, as Mitchell showed, vaccines have had the lion's share of funding for a long time. The science is moving forward. I think trials will be challenging in the future. So that might be a need in terms of community engagement. But, I think just really defining what is it right now that we need.
Thanks.
Yeah.
I think you're right there. The word community maybe, is probably overused. And for me, I see policymaker, Ministry of Health as also being community, a different community. And I do appreciate your point saying about the need to engage community. So I'm going to change my order of question and go back and forth. But what about the community leading the advocacy? We clearly, AVAC was born, correct me if I'm wrong, if Michel Leroy is here, to do HIV vaccine advocacy. You have evolved, like the virus has evolved. Go back to my earlier point. IAVI was funded to make HIV vaccine, and they have evolved too. We've got two large organizations that were created to develop an HIV vaccine that have evolved. And how do we recreate this grassroots activism for an HIV vaccine? Is it possible? Is there an interest? And how do we support that? Because I think that if there was as much noise coming from the community as we have heard from PrEP, we were banging pots in front of the Ministry of Health when I was in London. you know, how do we recreate this? Is there even, and I'm going to be more than a moderator, I'm going to be a challenger today here. Is there even an interest? And I'm not saying there isn't, I'm going to play the devil's advocate. Is there an interest? If there isn't enough interest, why isn't there enough interest? What can we do to stimulate this interest? What can we do to actually not look at the community as a recipient of research, but as a community that will say, hey, we need an HIV vaccine? Because I do not hear this message. Or maybe I'm deaf.
Thank you. So my name is Kyle Gordon. I'm an external relations project manager from the HIV Vaccine Trials Network. I think when it comes to building advocacy, one of the opportunities that we have is contextualizing choice. Speaking of the United States, I actually participated in HBTN 084. Was on long-acting CAB, not on the placebo, but when the study ended and the trial ended, like, it is a mess and a half for me to actually get long-acting CAB from my primary care provider because they haven't added it to the formulary yet. And one of the reasons why I say, you know, kind of contextualizing choice is certain segments of the community aren't going to take PrEP. You know, it's been around for the last decade. And if the uptake was where it was, you know, there will always be a need for a vaccine because the interventions that we have aren't broad enough at the local level, at the state level, country level, or the international level to kind of get us to where we need to go. And I think we also have another opportunity to infuse our HIV vaccine message in competing priorities. You know, I think one of my colleagues said that people have to deal with climate change, food insecurity, housing insecurity. But in the United States, if you overlay all these social determinants of health together, including HIV incidence, you're looking at the same map. You know, so how do we couple, you know, reproductive health and sexual health messaging along with HIV to where we can reach a broader audience? Thank you.
Thank you. I've got 3 people online. I'm going to do that in order. Ntando.
Well, thank you. Thank you very much. I think this is a great discussion. And I think largely we are the ones who really care about vaccine research. And I think that's an honest reflection that we need to, you know, to have, that we actually, you know, are keeping these conversations amongst ourselves. And however, I think the message is that we need more people who care about vaccines. We need more communities who care about vaccines. And I think, you know, taking the example of what Maureen is saying, that we now live in a world where we have a public that gets excited about, you know, things that are sensational, like we heard about this minister, and I think people then start to be hyped up by those kind of events. However, those are not sustainable. So we therefore need to find ways in which we can be able to sustain all of those. We need sustained approaches of building interest in HIV vaccine research and advocacy, which is exactly what I think was mentioned, that we just need to make a case for why we still need an HIV vaccine in the midst of all of the interventions that we have today. So we also have actually another group that I think the investment we have made over the years in engaging communities are those communities who have participated in all of the research over the past years, could be 30 or so. And that's really a group of people who have given their time, we've given, you know, we've supported their communities, who have supported the participants who took part in research, community advisory board members who have just been part of advocating for research. However, one of the things we do is to really forget about those people. And this is really a group that we can, you know, just really work with to be able to make more, you know, the case strong of why we need vaccines. Otherwise, generally, I think, you know, communication, messaging, engagement approaches should really move with the times. And the time today is artificial intelligence, and the time today is social media. Now, those are some of the very important considerations we need to have.
Thank you, Ntando, and a great point on engaging and building on communities that have participated in research, because they are the ones which actually, contributed to the research, understood the research, were part of it. Michelle.
Great, thank you, Roger. With the greatest respect, and this is super helpful to hear, but with the greatest respect, I think in many ways, I think you're asking the wrong question.
That's fine.
And maybe we need to think about, what are we trying to solve for? As you saw, Jean-Daniel and I think the colleagues from the VTN, people are enrolling in trials. There is an understanding that we need an HIV vaccine. So I worry about a lot of things. That's kind of my day and night job. But I'm not so worried that we need to beat ourselves over the back that and again, I'm not trying to say it's okay that AVAC and IAVI both expanded. I think it is quite good that we evolved, because the field's evolved. And so I don't think we need to be beating ourselves over the back and saying, oh my God, we don't have vaccine advocates. We do have vaccine advocates. And they're talking about some really important things. There was a series of conversations last week amongst advocates in Africa about local manufacturing. It wasn't only about HIV vaccines, but it was about vaccine manufacturing capacity for the long term, because right now, as you all know, we don't have an HIV vaccine to manufacture. But how do we get civil society and advocates and policymakers engaged, for example, with Afrigen and the mRNA hub, so that if we are lucky enough to have an mRNA-based HIV vaccine, that we'll be able to manufacture it in Africa, in Asia, in Latin America? That's the advocacy that's happening in very real time. So I'm actually not at all worried about that. We need to make sure it continues, that it's sustained and sustainable, but communities are really driving those conversations. And I think it's a really great point about the experience with cabotegravir. And I would want to say, it's easy to start making it sound like, well, PrEP won't work well enough, which is why we need a vaccine. I think that's a fundamentally flawed argument. Yes, PrEP is struggling in all of its forms, oral pills, vaginal rings, and injectables. But if we can't deliver those in the next 5 years, it's going to be a hell of a lot harder to deliver an HIV vaccine when we do get one in 7, 10, 12, 15 years. The best thing we can do right now as a global community is to walk and chew gum at the same time. If we can figure out how to deliver injectable cabotegravir with 6 injections a year, it will pave the way for a 2-injection lenacapavir in 2025 or 2026, which will pave the way for an antibody or a vaccine in the out years. And so I think we need to be really smart and strategic in our advocacy, that we don't try to undercut and say, well, cabotegravir won't work, which is why we need a vaccine. No, we need a vaccine because we need a vaccine. It's the best way for a durable end to a pandemic, period. In the absence of that, treatment, U=U, and PrEP in all its forms are going to be hugely important. They don't replace a vaccine. And we need to make sure that we don't throw PrEP under the bus, so to speak, because if we don't build the platforms for it in the next several years, when we do get a vaccine or an antibody, our work is going to be a hell of a lot harder. So we need to figure out how to work in common cause with these issues and not say that, vaccines are somehow unique. They're part of a continuum, and we need to build the advocacy for that continuum.
Thanks, Michelle. And with even greater respect, I do agree with you that some people do understand we need an HIV vaccine. But what I'm querying is, do all those who have, the access to the purse and all those who take decision really buy into that argument? And you are right, and we are going back to...
I would just say, look at the resource tracking data, and it tells you that even in the midst of those disappointing results, the existing funders are still there. And it's the Gates Foundation, it's the NIH, it's the MHRP, and it's USAID. Do we need other donors? Absolutely, but that's a different conversation, I think. And I think it's about making sure the science is fundable, is for me the most important thing.
So maybe my perception is tainted because here we live on another continent where funding is very much different. And as much as I agree, that the funding has been sustained, that the funders are committed, we need, as we said, we need more source of funding. And I was going to say, amazingly, we seem to go back to the conversation we had 10 years ago about the combination prevention approach. And I think nobody here is saying that PrEP is not going to work. But modeling recently published showed that, by 2030, it's not going to be enough. So the question is not, will it work or not? The question is, will it be enough or not? Therefore, we need other approach. And that's the way to support that vaccine advocacy. But I'm going to stop here. And there is Gaston and then George. Gaston is online.
Thank you, Roger. Well, my name is Gaston Devisic. I am the community engagement representative for Fundación Huésped, which is an NGO in Argentina. I would like to thank the AS for hosting this event, and I apologize for not being able to have met you in person. Yeah, it's true. I mean, even though it's really regretful that we may not have currently an HIV vaccine model close to a phase 3 trial, but this may be a great moment to start investing, as you all have said throughout the session so far, time, resources, and efforts to support the conduction of such studies when the time comes, particularly in regions around the world where such a thing comes with great challenges logistically, financially, and human resource-wise. For example, the clinical research sites here in Latin America and the Caribbean ended up having a very prominent role in the implementation of the Mosaico study up until last year, and I'm very happy to see Jorge Sanchez there, who can attest to this more best than anyone. So what can we actually do to prepare for the next best thing to happen, or to come up with that ourselves? What can we do to keep the conversations that we have started around HIV vaccines, so that we do not leave a message that we certainly do not want to be interpreted as a failure, of disappearance? And in that case, HIV is a victim of its own successes, and vaccines R&D is certainly not an exception for that. That is why and how advocacy is so important, so that we can contribute to the creation of the conditions for better, faster, and more diverse research to happen. And yes, messaging is important. Being part of the UNAIDS program coordinating board, I do hear a lot that we can't end AIDS today. And even though that we are knowledgeable of that being a fire starter for political commitment and for decision makers to put their money where their mouths are at, we also need to be mindful of not believing that ourselves. And the key to that is that we continue to think in terms of combination prevention, and that there's no such thing as a one-size-fits-all intervention, and that there might not be such a thing as a vaccine, but various vaccines, very much like we might not get a single intervention that can cure HIV as a biomedical, social, structural, and moral pandemic. Also, one would always want to highlight the purpose of these trials service and how important it is for them to be executed around high burden contexts for HIV around the world, not only because of the scientific value that the genetic diversity of the virus, but also due to the integrating approach to science and the need of conducting research in real life context, particularly those that, like my region or Eastern Europe, Central Asia, or even the MENA region, have seen an increase of new HIV infections during these last couple of years. So we need to continuously put our work into perspective. We certainly do not have time to be disappointed. There is a pressing need for us to develop new preventive strategies and to provide opportunities for people who may go in through a higher exposure to HIV that do not find in the current strategies with proven efficacy that we have sufficiency in them. HIV vaccine research is contributing to that now, and we always need to keep that in mind. I always bring this up because it doesn't stop surprising me after all these months. After Mosaico was interrupted in our site, half of our active participants still chose not to take PrEP, that meaning oral PrEP, even after the disappointment, the months of follow-up, the sensibilization to PrEP. So if that's not incentive enough for us to reconvince ourselves of why and when we need an HIV vaccine, I don't know what it may be. So thank you.
Thank you, Gaston. I'm going to give George away, but just before that, it's the third time we hear not everyone want to take PrEP, and I think that may be one of the message we would want to, put forward a bit more. George.
Thank you very much. I have 6 points to make. Let me try and get you through them quickly. The first one is so I'm speaking from the perspective of health management. So thinking of all of Europe, how are health managers making decisions? And I want you to think of a health manager as that person in the middle between where policy is made and decisions are made and where service is provided. In the middle is a health manager who translates each of those directions. And health managers are lots of different roles. They're sometimes called health manager, or they might be a nurse in charge of a ward, or they might be a policymaker. So they're very varied. So for us, we perceive this as a governance issue, obviously, of how you're governing these decisions. And I think of the example that I mentioned earlier of RSV. We did quite a bit of work on RSV prevention. And so we have looked at the impact of RSV on pediatric wards across Europe, on primary care, etc. And what is known and well established is that it creates a major burden on healthcare systems in November, December, January, that causes burnout and sick leave for staff, sickness in staff, etc. That's known. It's a known issue, and decision makers are perfectly comfortable with that because it's a known issue and we just manage it. And when we offered lots of different interventions, people had a very limited interest in it because it's a known issue. It's just dealt with how it's dealt with. And so there was limited interest. Although there's better prevention options on the table now, we'll see how those are engaged with by different health systems. But we do know that just alone, that argument, in fact, is it John? No. Your name? No. Tom, sorry, Tom, you made the good point of when things are cost effective, like invest now, save later. We also know that doesn't work. We know that investing now in this current government will pay off later for the next government. So that doesn't work for governments. We know that also very well. So we know that financial arguments have to be done very carefully because they don't work as well as they used to work. So next, that has me thinking of HIV exceptionalism. So HIV has made itself exceptional, and that was important for HIV, and that helped us to improve the way that we responded to HIV. And then it stopped helping us because now it's not that it's in the too hard basket, it's now in the too specialized basket, which means decision makers don't want to touch it because it's too specialized and it feels like it's dealt with. Now, if you look at Europe, we have the EU Beating Cancer Plan. It was a very surprising plan to come out because it was so disease specific, but what it does very well, and if you take the time to read it, and you need the time to really read it, it's not just about disease, it's about air quality, it's about our lifestyles, it connects to the EU Green Deal, it connects to the broader policy priorities of the European Parliament. So it was a very cleverly manufactured piece of policy work that touched on everything. They showed cancer being relevant to everything. HIV is not understood in that way. HIV has been promoted as being so specialist and so exceptional that when decision makers look at HIV, it's in the too specialized basket, not the too hard basket. So I think an opportunity for us is in making it more accessible for decision makers to do something that changes it. And then I think of the prevention paradox that we have in HIV. And I worked a lot before health management. I specialize in HIV and I worked in HIV community organizations. And so Gus, I think you made a mistake. You accidentally said UK, and I think you meant to say England. So England took its government to court on PrEP, but Scotland didn't. And I know that because I implemented PrEP in Scotland, and we had great success there. We became the first country in the world to have it fully integrated. So there's lots of good success stories there. So if we really focus on how did that work, might be some lessons learned there. And they weren't easy success stories. They were very hard success stories. And what we learned in that PrEP one was financial arguments didn't work. But other things did work and we got it implemented. Was the environment ready for PrEP? No. Was there a community group advocating for PrEP? No, there wasn't. But when we went digging, we found, we listened differently and we found a lot of answers. And instead of us telling the community what we wanted from them, we listened to them and they gave us answers. You know, it's such a cliche, but it really worked. Now I'm nearly finished, that's point 5. So on the prevention paradox, when I was working in HIV, we'd have case studies like this real one, where a young person went to the HIV clinic, terrified about having acquired HIV, seriously distressed about it, was very physically unwell and was taking time off of work and had become socially isolated, and went and got tested for HIV and tested negative. And and then was past a window period and tested negative. And what the nurse said to him was, if you test positive, we can give you a lot of support. If you test negative, goodbye, we don't want to see you again. So I didn't follow that case study any further. I don't know how his well-being continued from there. But what that paradox tells us about is we have no space for people who are interested in prevention. We get rid of them as quickly as possible. We don't want them in our system. We don't want to know about them. We get rid of them quickly. So then if we wanted to develop or give space for an activism of people interested in prevention, we have to make space for them. And the way we design our healthcare system has zero interest in them. We don't want them anywhere near us. So we also defund their organizations. So, all of the community organizations that used to exist on, I'm thinking of gay men's health as just one example. I recognize there's other key groups. They are disappearing. Their funding is going. So how are we creating the space for these communities to mobilize and have their voices heard, or needs further thought? And my final point is that one. That's it. I just made it. There you go, number 6. Those are my comments. Thank you.
Thank you. Great point of the creating the space for actually having the communities and the organization to support and do some advocacy. And I knew GMFA because I was a volunteer. And again, to show the evolution, and it's not being critical. GMFA stands for Gay Men Fighting AIDS. When I was volunteer there, I think in the late 2010, the motto disappeared and it just became GMFA, and it became a kind of a One Health approach to sexual health of gay and beyond men. So I can't express that, because it's too many words, but it becomes more holistic. And that may be also a way to, and I'm opening, that's a suggestion, opening the way to the advocacy to HIV, for HIV vaccine, go beyond solving the problem of HIV, but solving the problem of, as we heard earlier, AMR, solving social problem, but also making a contribution to society. And I want to make sure that the scientists have a say in that discussion. And we heard earlier about the need for arguments and for support. And we have words like, we heard words like TPP, like having milestones. I heard the menu. What does the scientist think they could contribute to support the advocacy effort without going into the hardcore science? Menu, is that a roadmap? Is this feasible? And have we identified the scientific contribution to the advocacy? Because as I started, I said, good advocacy we build on good science. What can we take from the science that is understandable? My first question, that can be presented to the right people and that will convince them. Do we have these arguments? Do we know how to convey them? I'm just opening that, Jorge.
Wanted to comment about community participation in the Mosaico trial. There were 4 countries in Latin America participating in Mosaico, and these countries didn't have a vaccine trial before in HIV. Only Peru had a previous one, but was 15 years ago. So there were naive countries in terms of vaccine advocacy for a vaccine trial. And all 4 countries did perform very well. And I think what happened is having community people in our research teams were key to get participants for the 4 countries. So there was no country that was lack of interest from the community. And I think it's feasible to get participation to vaccine trials if we reach the community with at the same level, not going from the scientists to the community. So going from community to community and get participants able to, or people willing to participate, able to reach the clinical research sites. So I think it's feasible. It's happening in countries that had no experience doing vaccine trials. So I think that's feasible to do it.
Thank you. Any other comment in the room or online on what can? Gus, go ahead.
Yeah. Okay. I'm going to try and get to where we are now by talking about how long I've been here. because I'm old enough to actually remember gay activism before HIV ever existed. I was in Gay Switchboard and was a volunteer there and didn't expect to be paid to do anything. I was a bus conductor to finance my activities at Gay Switchboard. So I don't think community anger and activism has ever depended on funding personally. I think it depends on a community understanding that is under attack. And HIV community activity started with a community that was dying, advocating for itself because nobody else would. And it's been used as the first time the sufferers from an illness have ever been the prime movers behind the developments against that illness. So that's what started it. You know, all the ACT UP demos, all the sort of blood on the streets. I wasn't there. I was too busy surviving at the time, but I was there at the Durban moment in South Africa because then that anger moved to South Africa. To Africa, and that was incredibly inspiring to see. That we were told that civil society simply wasn't an African tradition. Well, we certainly proved that wrong. Right, but then we realized that we had to start understanding the science and building alliances with the sciences in order to get out to sharpen our messages. And some of the ACT UP generation developed alliances with people like Anthony Fauci, because they knew that what they really needed was a strong sense of strategy to, like, this is where we need to get to, okay? And then we go into prevention. Well, I think PrEP in a way is more like an ACT UP issue. We can see it works, and it's simple. You give people a pill, it protects them against HIV. And I actually slightly disagree with you that I think there is terrific PrEP advocacy around. In the UK, there has been, in Scotland, in England, but also in Europe. You know, I was the coordinator of the 2 PrEP in Europe conferences. There's been amazing work done by people like the Love Tank and so on. There is community activism around PrEP. It's not there in every community that needs to have it. With vaccines, we're more at the making friends with Anthony Fauci stage, I think. The most interesting things I've heard this afternoon have been my conversations with some of the scientists, like Dr. Yu over there, about the specifics of vaccine development. And there is so much I don't know, you know? I mean, the science of vaccines is just way, way more complex than the science of antiretrovirals. It's like, you know, the quantum physics of medicine. So we need, I think, as a community to talk with the scientists who are developing the vaccines to get a much better understanding of where we're both trying to get to. And that's been the most positive outcome for me of this particular meeting. I do want to add something about, I'm not going to be negative, but about the UNAIDS end of AIDS target, because I do worry that this was too ambitious, and that it might end up being a negative factor in the broader global community's understanding of why we still need a vaccine. Because although PrEP and U equals U and everything else is certainly necessary to bring HIV levels right down, it is not sufficient. And I know that because I was just credited as a co-writer on a study of the UK epidemic, which forecast that we would never essentially get to that UNAIDS target, not for at least decades, because it just doesn't happen that way without an additional intervention such as a vaccine. So although I think it's useful to have aspirational targets, I was a supporter of 1990. I think I do worry that it's been a break on some of the perception that a vaccine is still very much necessary if we're going to drive HIV out of the system altogether. So that's really where I want to get, where I want to finish. Thanks.
Thanks. And we are reaching time, but I want to ask that question to Prof. Lu, because you escaped the room when I was asking it, and it follows up from what Gus said. As a scientist, what do you think you can contribute to strengthening the advocacy for HIV vaccine? In a brief response, how do you think scientists can help?
I feel there's a huge gap among scientists. Let's say there's a community, it's a very broken community, and the outside the non-scientists community advocating or general public. Eventually, they may be the the volunteers or the population will receive HIV vaccine. I think that there is no communication at all. I feel that's not good. When my vaccine now reach a phase 1, phase 2 level, I feel this has become even more important. Not only for the older reason you guys discuss here. I don't think in this short, I have any experience or to say anything, but always in my mind, I think that's what I have been trying in the last few years, to reach out to people in South Africa, to South America, to other places. I think we have to work together to understand each other, to explain the science, and also hear back what the community needed. Actually, if you are in a company, industrial, how do you develop your clinical trial? There's some arms, some stop groups. You really need such information to help you. Most people probably don't realize that. This is not something a basic scientist will think about. But as a product development in a company, they look at this kind of thing all the time. Just for this selfish reason, I think we can talk, but also more importantly, to get their support and to see whether our vaccine will have any value and how soon we can deliver that. How can we change along the pathway, say a long process? How can we improve that? I think all this, we need a communication. So I fully, that's my personal feeling.
Thank you. Conscious of time, I'll just ask, like we did in the previous one, very shortly, the theme of this meeting was not business as usual. So thinking about advocacy, a one sentence, not advocacy as usual, what would be your key message? I shall start with Maureen, if you don't mind, or you want me to start by the other end of the room. Maybe I'll start by the other end of the room. George, not advocacy as usual. What's your key message?
No.
Use the EU Beating Cancer Plan as an inspiration. Let's draw from that to see how can we represent HIV vaccine to make it feel differently relevant to decision makers.
And for me, I think it's widening the circle of trust and diversifying who we include.
Mm-hmm. A minute to think.
No, I mean, do more of what we do well, but also think about new approaches. Don't just do the same.
I would say scientists have to learn how to speak with people. Because I just realized that also we started the Core 07 and it's a therapeutic trial, and there is a great response, but sometimes they look at me like, what are you saying? What is that? So we have to really learn how to speak with people about science.
I think someone said it earlier, good advocacy builds on good science. So how are we ensuring that us as stakeholders, but also, kind of different people along the pipeline ecosystem are engaged in the science so they have a better understanding of what it is that we're trying to do with an impact of a vaccine to relay that to a general audience?
Yeah, I had similar comments to make, that clear communication is really critical and that we all work harder on that.
I think a way forward here would be to ensure that there is a continued interaction between the advocacy arm with the scientists, so that as the scientists interact more with the advocacy team, they come on board, understand the process, appreciate that beyond science, you need to communicate with the communities. Yeah, thank you.
I will echo what has been said already, but it is for me hearing what has been discussed today. It's really about having the proper kind of communication. So from scientists, and in both ways actually, from communities to scientists and from scientists to communities, and also expanding a little bit. I found that very interesting, the conversation that we had about what is and is not communities, but it's not just people living with HIV, it's much broader. So it's targeting advocacy to the various stakeholders and not seeing that in one way, direction and communication. Thanks.
Yes, as I was saying, better communication between the scientists and the community researchers. That's partially responsibility of the scientists to explain all this complicated stuff about, you know, germline targeting and V1, V2 loops, and we don't know what that means. But also it's our responsibility as a community. When I said I was going to this meeting, I had two EATG activists who both said to me, oh, well, we won't have an HIV vaccine until we're both dead. And I think that's wrong. Potentially, or certainly we need to kind of overcome that kind of pessimism amongst the community as well. So I think it's partially our responsibility to keep up the interest in the possibilities of a vaccine.
I would say that we, the scientists, need to learn of successful stories that have happened in the past, and also for non-successful stories, because we cannot make the same mistakes in the future.
I think scientific community probably underestimate how much the non-scientific community already know. And the scientific knowledge, like our friend here said, you know, germline targeting or anything, they know a lot. So being a scientific community, we should really improve ourselves. Otherwise, you cannot even tell other people because they know so much already. They are completely different from 10, 15 years ago. Sorry, I'm saying a little bit longer. I visited South Africa, for example, as an example, many other African countries. I was truly shocked. People are so well organized, well trained, very knowledgeable. They're waiting for the vaccine. So it's our failure, it's not theirs. So that's what I learned, truly. So I think the language, the wording I was going to tell people 10 years ago, no longer useful. I have to re-educate myself. That's my finding.
Yeah, I fully agree with everybody's opinion. I think the better communication, learn what the community need, and then you know, as a scientist can better provide the most important information to them.
So I think we need to dig deeper into the science, especially as an advocacy community. And I think we need to have, I mean, I think this is building on a lot of the comments that have already been said, but I think we need to really take an honest look at where we are in the state of vaccine really research and like basic research right now, and come up with some clear, very clear messages about why there is hope and promise for the way forward.
Yes, so I would say that you know, I think the scientific community would do well to, you know, just listen to the, you know, to the communities, quote unquote, including the trial communities as well as others, and especially the people just on the ground, because I think there are very many lessons that they can teach us, including scientific lessons from the observation of HIV over the last, you know, 40 years. Perhaps there's some clues that we would be missing by focusing on our own research without going back there and really trying to see what it is that is going on the ground. Thank you.
Yeah, I think everything has already been said, but I think I'll just repeat that I think we need to continue to create more opportunities and spaces where communities can engage. I think most of the times we tend to limit community engagement, maybe in the form of a club at a clinical trial site. But I think there are more spaces where we could bring in a community. I think we heard from Mitchell a bit about some of the conversations that advocates have been reading around vaccine research. So I think we need to create more spaces, even at the highest level, I think, where we can bring in communities and they share their insights and contribute to the conversations that we're having. Yeah.
Thank you, Maureen. And we have a comment online from Jean-Daniel. No.
I think, sorry, I think it's quite different for me because first of all, I'm a medical doctor, but I will say that the first thing for me, when you are a scientist or medical doctor, the first thing to begin with is rather what you want to achieve, but why you want to achieve, and try to express first with your heart, not with your brain, and you will succeed in your advocacy, at least for the general population.
Thank you. So I'm going to end that session and complying with Tom Hankey's order for optimism, because I really believe we have had many more success than failure. We just don't talk about them. And whether it's RV144 AMP or the development that supported, COVID-19. So I'll end by quoting Shakespeare, which is not very common in science. And with the fifth, once more into the breach, my friend, once more into the breach. Over to you, Marlene.
Thank you. And thanks a lot for Roger for moderating the roundtable discussion and for all of you who've been participating. Now it's time for our rapporteur session. We've been discussing a lot of various things. It has been a very rich conversation, and we're just going to have a very short summary. So we leave this room today with the key take-home messages. And it's a great pleasure of mine to introduce our two rapporteurs. We've been hearing about them quite a lot today already, but let's introduce them very formally. So our rapporteur, our first rapporteur is Shan Lu. And Shan Lu is a physician scientist and global leader in vaccine science with significant experience in managing large multi-institute biomedical research and product development programs in the field of novel vaccine. He is director of the Laboratory of Nucleic Acid Vaccines in the Department of Medicine, University of Massachusetts Medical School, USA. And our second rapporteur is Maureen Luba. Maureen is a Malawian global health practitioner, currently serving as program manager at Cooper Smith. She helps lead the organization's effort around strengthening health information and data system in Malawi to improve health outcomes, working closely with government and other stakeholders. And she's also working with IS for our community-led monitoring initiative. So I'm going to give the floor to Sean and Maureen to tell us about the key discussion and highlights of today's meeting.
All right, yeah, I'm happy to go first. So I was covering, of course, the second part of the discussion, which was focusing on the reventing the HIV vaccine advocacy. So we're doing the report back in reverse. But I thought part one of the meeting provided such an excellent framing, I think, of the discussion that we just had, by first of all, having that reality check from Dr. Vincent in terms of where we are with the pandemic, the progress that we have made so far, the successes, but also the challenges that persist, especially around new HIV infections among the younger generation, considering the demographic transition, which could potentially reverse the gains that we have made in the HIV response, according to Dr. Vincent. Of course, as usual, there was no way you could have this conversation without reflecting on some of the lessons that we have learned from COVID-19 vaccine development and what we should be advocating for in the HIV R&D space. I think the resounding message from part one of the discussion, which I know Shanlu will summarize, was that we still needed a vaccine, even with other interventions, PrEP, available. But I think we need a vaccine, something that could provide that long-term protection, that would allow maybe fewer interactions with the health systems if it's available, but also provide access to the most remote parts of the world. I think overall it was encouraging to see such great optimism. I think we are all optimists, as Mitchell said. That's the reason why we started talking about an HIV vaccine. Otherwise, we would have given up a long time ago. But I think one key challenge, of course, that was highlighted from the lively roundtable discussion that we had earlier on was that, which I felt is critical as we are thinking about reinventing or rethinking our advocacy strategy for HIV vaccine, is that there seems to be no clearly defined mechanism or commitment. I think this came out from government and maybe private sector in terms of support for HIV and D. And I think it's something that we need to keep thinking about. Of course, the second part of the discussion started with a presentation from Mitchell Warren on the HIV resource tracker. Where's the money? And he showed us, of course, where the money is. I think in his presentation, he kind of like reminded us where we're coming from, how far we've come in the field, and where we're going, but also what needs to be done as we continue this journey. I think he said, yes, he reminded us that the conversation that we've had today, the commitments that we are all making, it's what we already committed almost 20 years ago. So I think these are the right conversation. We need to continue having this the conversation. Of course, he said, yes, we need the money, right? The money, we need more money. But also, I think he reminded us by, I think in this presentation, that this is such an incredibly funded enterprise. I think there's been a lot of funding that has been allocated to HIV vaccine research, and something that I don't usually personally reflect on our supplies. I think to hear that, like the TB vaccine field is actually one of the underfunded, and we know there are other, HIV prevention trials or products that still remain underfunded if you compare to HIV vaccine. So it's important to recognize that, we are talking about a field that has been incredibly funded. I think in his presentation, of course, he showed us how there was such an increase in funding for HIV vaccine research following the creation of the HIV vaccine enterprise. But I think the key message was that, yes, as much as we need more resources for HIV vaccine research, I think we need to start thinking of in what places, areas where additional resources could add more value. We need to start developing a menu, right, of novel intervention strategies where additional resources could actually add more value. So it's not just about asking for more money, but really identifying areas where, those additional monies could add more value. We also kind of like, reflected, took us all to reflect on what we have in the pipeline, to say there's quite a number of products, of course, in the pipeline, which does not necessarily mean that we don't need an HIV vaccine, but only it raises the bar higher of what a vaccine would actually look like. And I think it's something that we need to continue, discussing. We had, I think I would say, also a lively roundtable discussion facilitated by Roger. And I think a few things, key takeaways that I got from the conversation that we just finished was that we need to continue to invest more in or create spaces where communities are meaningfully engaged, and that we need to continue, especially for our scientists. I think we need to make sure that we need to continue to explain, to break down the science, so that communities do understand, the different products that are being studied, even as complex. I think we all acknowledge that the HIV vaccine research continues to become more and more complex with a number of areas. I think as we're trying to think of novel strategies, I think the more complex it becomes, but that doesn't mean that we shouldn't be putting in more effort to explain that science so that we are moving along with the communities. I think there was also, yes, just a reminder that I think sometimes we tend to define the community more broadly, right? But I think clearly define what the community that we are talking about. When you're talking about HIV vaccine research, what is this community? What is it that they want? Really, taking time to listen to the communities, to spend time with the communities, I think would be is key as we're moving forward. And I think Roger asked the question around like what is there activism? Does it even exist? HIV vaccine activism, is it there? And we heard, I think, from the different comments from individuals from the roundtable discussion that HIV vaccine activism is there, and that communities are really leading critical conversations around the need for an HIV vaccine. Communities are actually waiting, for this vaccine. And I think we heard from the story that I just shared about, Kabele in Malawi and the excitement that people had when they heard that this Kabele is a vaccine. People were really excited. So it still shows that, people still have hope, and they're still waiting for the vaccine. And of course, I think in summary, I think if I can summarize, I think from the roundtable discussion, I think some of the key takeaways is that I think there is need for us to keep pushing for combination prevention. Yes, we need an HIV vaccine, but we also need to recognize that we still have other products that are available, that are efficacious. We have Kabele, we have Oloprep. So let those that can use those, it's about choice, right? It's about providing all these products in the context of choice. Messaging is key. I think that also came out. Messaging is key, especially in terms of, yes, we need aspirational targets. We had a conversation around the targets that UNAIDS has been leading us in setting those targets. Aspirational targets are great, but I think we still need to create room for a vaccine. So yes, there was also, I'll be finalizing, there was also, I think, some insightful, comments in terms of, yes, as we're having this conversation around HIV vaccine R&D, what are the implications on healthcare system? I think it's something personally I also haven't reflected on, that, sometimes maybe financial arguments do not have to be done, carefully, right? Sometimes they don't always work. I think it's something to keep in mind. HIV exceptionalism, I think it's something that I haven't thought of. Thanks for bringing that up to say, I think it's like we have boxed HIV, we have put HIV in a box as if it's something special, and that we need to make it more accessible. But also, yes, I really like the point around, I think sometimes the way our HIV programs are designed, they are more designed for people who are, right, HIV treatment. It's like we are there, we are testing so that we can find people that are HIV positive, and then we give them, we put them on treatment. But I like this new concept, which I think EVAC has also been using, test and reach, right? Like you're not just, you want to reach everyone, not just only those that are test and connect, not only those that are HIV positive, but we also want to make sure that those that are HIV negative are put on funding. So yes, I think moving forward, all you need is, we need to sustain the current levels. These are some of the key takeaways from Mitchell. We need to sustain the current levels of investment. We need to diversify, of course, investment beyond the US government. But also, yes, I think coordination, global coordination and collaboration, this also came out from the first part of the discussion. We can't work alone. We can't work in silos. We need to strengthen collaboration, but also strengthen political commitments, I think, will be needed if we are to, get to find an effective vaccine. And we need to continue, of course, to listen to the communities, I think, as I said earlier on. Otherwise, I think, I think the resounding message was that, yes, I think we are all optimistic that even though I know we've come a long way, even though, the field is becoming more and more complex, but I think we are all hopeful that soon, maybe even sooner, we will be able to find an effective HIV vaccine. So that's what I captured from the conversation. Thank you.
Thank you, Maureen, for your wonderful summary that make my job much easier. So I would just first want to thank IAS for organizing this, because the continuous support for HIV vaccine development from IAS will be very, very important. Even though there are other organizations, funders, but without IAS to actually run that, it will be impossible for all the other effort. So I think I personally really appreciate and thank you for that. I also think this program is well designed, thanks to the enterprise staff and generations leaders, including Roger and Gabriel. You guys are continuing, even though you are not exactly in this role, but you have contributed and done a great job as the chairs for 2 sessions. Really, I think I want to thank you on behalf of every participant. I think the speakers, first two, gave us first, Vincent, a broader view of HIV vaccine approach. And Sheila gave us particular the most hottest stuff, mRNA vaccine from African country point of view. And both of them actually being the speaker here, showing the growth of younger African scientists. I think that is more important than anything. This is very exciting to me. And also, I just want to comment that Vincent, I think he paid special attention to the neutralizing antibody and T cell. He touched a little bit ADCC and other thing. I wish he could have expanded a little bit more as just a friendly suggestion. I think, Sheila, the talk is excellent. When I first look at your title, I was a little bit concerned. You say, hold your enthusiasm. Is something wrong with the mRNA vaccine? I was hoping to find out, but I failed. Thank you. But also, I understand the challenge. Many African country or Asian country try to develop an mRNA vaccine supported by WHO. Now they transfer tech to all of you guys. I think the in vitro laboratory work is easy, but the GMP manufacturing will be a huge daunting task. So I wish you the best luck, and hopefully we can see more spreading of new technology in many low or middle income countries. So from that, I see also another sign of hope. I certainly always enjoy longtime leader of AVAC, Mitchell. He gave us another wonderful summary. Some of the data I could not find other places. I took secretly a lot of pictures, but I know I can find it from the public available recording, because when I write to investors or others, this number are very important. So tell them what is the funding status, where they come from, what's the gap there. But I guess the take-home message from Mitchell is we do have a lot of money. The question, how do we spend it? The other thing I did not pay attention before, but I learned from him, is actually after the so-called failures of multiple big trial, the total funding pie, would that reduce? If not, how do we reallocate? I never thought about that. I don't know who will be responsible for that, but that's a good question for big, players here. So I think people should think about not to waste the valuable money we accumulate and put into the curve, don't get a big drop, and the use wisely. So that's my learning today. So I think Maureen talked many of others, so I will focus on the roundtable one. I think we all agree this is very exciting and lively discussion, almost the level of debate, which I think we need. I have participated in many enterprise webinars and other. Most are well structured. So we have the leaders give us education, but less likely, like this open-ended discussion from the people from very diverse background, from different country, region, different scientific or community background. I think this is what we really need. So the roundtable started with the chair's very challenging question. Why COVID succeeded, but the HIV is still struggling? Certainly, we talked the difference of virus, many other things, but some lesson we should learn from the society, how to support that. Then we talk about difference among the three big challenge vaccine in history in human being, the societies, malaria, TB, and HIV. I think there's a sense we may see the progress of each of them. That's my personal view. I have the luck recently attended quite diverse meetings. I predict in the next 5 years, we may see more exciting news among all three, including HIV. Let me hope, keep our optimism. Then we have a little bit of discussion or debate on the role of biotech versus pharmaceutical company. I hold my view. Pharmaceutical will not invest not only HIV, even emerging infection, because the nature of a business cycle for a disease cycle. So we should not blame them. We should work hard and create a product they can invest at the end. From this end, I think Moderna, I personally think we should not underestimate this biotech. because now all of a sudden we had new biotech made money from COVID. They actually want to invest in HIV. So see, this is a cycle fully come back. Mitchell summarized it very well. I was going to say HIV vaccine field trained, educated many other people, neutralizing antibody assay, the fusion domain, so many things, including the HVTN trial network, led to the success of COVID. But at the end, something will come back. So I'm very also optimistic on that. Then we talk about how do we select the targets, T cell versus BNAbs. I think that the consensus also we see they are both important. Also, Tom and Barhan's all reported some progress on that. So I think we should look forward to this progress. And also mRNA vaccine seems the focus at this time. People talk about how to move it faster. And certainly there are some safety issues. We should not ignore that. And then we talk about, people talk about release manufacturing as the same thing I mentioned earlier. When mRNA vaccine go to HIV, go to many countries to manufacture, that issue may be more important. But the same thing, we come back, talk about discovery medicine trial. People feel this may be still useful, may generate some information for us. Let's think about it. Let's wait to see. Also, I think we have a participant comment, the immunology part. Some people still believe the sterilizing immunity. I personally agree. For HIV, you don't have sterilizing immunity, you don't have HIV vaccine. So people say the mutation is important for HIV vaccine. I feel sterilizing immunity is actually higher bar than any other vaccine for HIV purpose. People talk about what is the immune system. We still need more. And also people ask, should we follow the same track, same current system, but what are the other systems? I think people raise that question, but we don't have an answer. We acknowledge there's a knowledge gap. The question is, certainly we look forward, we may need more combination vaccines, but where to exchange ideas. We talk about whether still the value of enterprise, what the role of enterprise as a business manager or as a scientific platform. And this question is open. I don't think we hear a clear answer. I know actually the immediate decision probably not made by enterprise itself, not by people in this room, but I think we should feedback to whoever make a decision. I personally will say enterprise is very important. As the first, what is it, 10 years, 20 years history, 20 years, the first paper by the dominant people. I think the same reasoning still exists. I don't see that should change. So if I ask my personal opinion, I think we should keep the enterprise. But on the other hand, we have participants here asking a very good question. From a community point of view, it seems the whole field is very disorganized, poorly managed. So how to find this mechanism, systemic management approach, use industry approach to manage HIV vaccine, both policy, priority, and technology. And that's very important. I think that's a very important suggestion for either enterprise or whoever want to be involved in the policy decision process. We talk about whether there's a post-mosaic trial, pessimism or optimism. I think at the end, optimism take over. We are dominant because otherwise we will not sit here. We will just close the door or close enterprise. Hopefully that is found. And also we talk about the lack of a system for efficacy trial. I think it's not just a funding, it's rather the process. To me, it's the process. Selection criteria, and who will make a decision? That's very important. I can see people recently already set, so what is the efficacy standard? 50, 70, 80. I question, how did you get those numbers? Based on what analysis? Based on the whole population benefit or based on immunology? I think this is something we need to debate. I don't think this is the time to make abrupt decision on that. Then we will limit the science and discovery. So I think people ask questions, what is the system for efficacy? I think the roundtable is very lively. I will not go through them, and I cannot remember of them.
But I think the key message is still is to keep optimism and keep open-minded, and also to look at certain topics that we have not paid attention. And finally, I agree with my colleague from South America. This is a very good meeting, and we should have more like that. So that's my summary. Thank you very much.
Thanks a lot, everybody. So now it is time to wrap up, and I would like to thank very, very much our speakers, our moderators, our rapporteurs, and all the participants, both here in Geneva with us in very cold Geneva in person, and all of you who are also joining us, who have been joining us online. And also massive thank you to the IS team who has not been very vocal today, but working tirelessly in the back of the room and for many days and weeks at the IS. So big thank you to Ashley, to Julieta, and to Julia for their hard work on this. They deserve a round of applause, I can tell you. I would like to inform you that a summary report will be circulated. Looking forward to seeing the recordings, but also reading all the reports. And I wish you all a good evening, afternoon, or morning, depending on where you are. And hopefully we will see you again very soon. Thank you.